response. Hormone signaling, sugar metabolism, and ROS. This also work to build a validate a regulatory network of transcription factors and their target gene that drive degradation, meaning the look beyond single gene and consider how genes regulate each other. During the bamboo degradation of muscle bamboo shoots, there were reports rupture protozoan cell and reduced cell wall thickness, along with accumulation of sucrose and RS. This gene signal is structure damaging and metabolic imbalance as the degradation process. This tells us that the degradation is not just superficial symptom and involve deep tissue damage and metabolism inside the shoots. This observation comes directly from the study abstract, which summarizes these structures and metabolic change during bamboo shoot degradation. The transcript analysis, looking at the gene expression, highlights that gene relate to plant hormone signal transcription, sugar metabolism, and ROS are significantly involved in the bamboo shoot degradation process. In other words, multi pathways interact. One is hormonal signals, sugar handling, and stress mechanism. This supports the idea that degradation is coordinated biological response rather than a single cause. Understanding which pathways are involved help understanding why intervention, genetic or management might be effective. Going deeper, the study identified the co-expression regulatory network of synthesis associated transcription factors, especially 3 genes: ICG39, WORKY22, and WORKY75. These factors are primarily located in the protozoan of vascular bundles. The varieties were anatomical damage were observed. Importantly, functional assays show that WORKY22 and WORKY75 directly attack active ICG39 by binding to its promoter. They created a direct molecular link from transcription factor to a key target gene-driven degradation. So the study advanced our understanding by not only describing what happened during the degradation, but also revealing who the molecular regular are and how they interact. In summarize, the machine mechanism in the simple flow. Environmental stress and internal costs lead to accumulation of RS and sugar in protein cells. These attractive worky transcription factors, worky 22 and worky 75, they binding and attractive SAG39, a senescence-associated gene. SAG39 targets process that break down cell structure, lead to shoot degradation. This mechanism change give us a clear note. LS working factors, SAG39 for future research or practical intervention could focus. Why does all this matter under climate change? Climate change affects growth in environment potential in increasing stress, disease, and degradation risk. If bamboo shoots degrade more frequently or more severely, food supply, local income, and industry settled could suffer. By understanding degradation mechanism, we can better predict when degradation might occur and take steps or prevent or delay it. Strengthen the resilience and productivity of bamboo forests as carbon sinks and economic assets. This research suggests several avenues for sustainable use. Molecular markers and breeding. If we can identify or develop bamboo varieties of lower sensitivity to degradation, we can stabilize yard salt genetic selection. Cultivation and management practice that reduce RS accumulation or sugar imbalance. Perhaps salt target nuisance management, environmental control, and timing of harvest could mitigate degradation risk. Biotechnology and regular knowledge, understanding the working SAG network open doors to more advanced interventions. But this need careful to ecological and plastic evaluation before accumulation. Any strategy must be adapted to local ecological, social, and economic context to truly be sustainable. The potential impact are quite broad. Increasing yield and quality of bamboo shoots benefit food security and income for communities relying on bamboo. Reduce resource waste by minimizing the percentage of shoot loss to degradation, strengthen the reliability of bamboo material supply chains, support carbon neutrality, goes by maintain health, produce bamboo forests that continue to fix carbon over the long term rather than decline due to degradation. In essence, healthy bamboo shoots today translate to more sustainable forest and economic tomorrow. Look ahead, this clear next steps. Investigate how degradation dynamic vary, understand different environments cultivation condition and management practice, studying interaction with other gene or pathway beyond the working and SAG models. Field trials and demonstration, test management practice or select bamboo lines guided by molecular marker in real plantation, evaluates cost and outcome for farmers and managers. Cross-disciplinary cooperation bring together molecular biology, ecologists, and soil economic express to translate molecular insight into practical guidelines and policies that suit diverse regions. In summary, bridge basic research and real needs, connect molecular discovery to potential questions faced by bamboo grower, forest manager, or policy makers. In summary, I want to thanks to the research team, organizer, and funding source, and for this work and today's discussion. Thank you for your listening. I'll welcome your question and thoughts.
Martin, the field results of the sequence. And you show the picture of the sequence. Okay, so in your conclusion, you start both working 22 and working 75, both activate the downstream SAG 39, right? But in the picture B, you can see it seems that it's decreased expression of SAG 39.
Yeah.
So why you say bioluminescence is active expression?
So your question is why is decrease?
Bioluminescence can active, but the new result is decrease.
Oh, because the working 22 the expression is decline. The working 22 the expression is uh-huh.
I think the conclusion is that they should have some error in that. Because it seems that
Yeah.
what we do is decrease the expression of SAG 39, right?
Yeah.
But 75 is actually.
Yes. Okay, thank you. No. Okay. Thank you, everyone.
Certainly, the bigger is the human dragon. Recommend bamboo flowers of my model, and it has an annex. I'm not sure if it's with that information. And it's something random.
Good morning, everyone. My name is Enzo Battagliambula, and I am an international student of Zhejiang Agricultural and Forestry University. And thank you very much for taking the time to attend my presentation. Today, I will be presenting the research project that my team and I have been developing on, which based on machine learning driven development of a bamboo callus growth model. To guide you through my talk, I have divided the presentation into 4 main sections, which base of research, background and significance, objective and technical roadmap, process and result analysis, conclusion and future work. This section in the research background and significance. This section introduced the context and motivation of the study. It explains why the research topic is important and what scientific or practical problem it aims to address. Callus formation is a key stage in bamboo tissue culture, directly influencing regeneration and embryogenic potential. Callus volume serves as an important indicator of growth, cell density, and regenerative capacity, with dense and robust calli showing higher organic potential. However, volume estimation in a sterile culture is challenging due to the irregular 3D shapes, fragile tissue, contamination risk, and variability from manual measurement. Measuring bamboo callus volume is crucial because it indicates growth and regenerative potential, helps optimize culture condition, and enables non-destructive monitoring for accurate, high-throughput research. A non-invasive digital system allows large-scale callus monitoring while avoiding contamination, paving the way for automated, fast, and objective machine learning-driven plant tissue phenotyping. This research develops a non-invasive automated system for callus monitoring that integrates segmentation, volume estimation, and quality assessment to reduce manual labor, minimize contamination risk, and ensure consistent measurement. By combining precise volume evaluation with morphological analysis, it enables objective and assessment of regenerative potential and support better decision-making in bamboo tissue culture. Overall, the work builds a scalable foundation for high-throughput phenotyping and digital monitoring of callus growth. Research objective and technical roadmap. Here we define the main goals of the research and the technical approach used. We also outline the roadmap, showing the steps, method, and guide the study. The project aims to develop an intelligent system that automatically monitors callus growth, reconstructs 3D volume from multiple 2D images, and evaluates morphological features like compactness, surface irregularity, and texture to assess quality and regenerative potential providing objective, reproducible, and high throughput data while reducing manual labor and errors. The workflow includes building a controlled image dataset with a real and synthetic sample, evaluating deep learning models such as MaskR-CNN, UNet, and FCN for accurate callus segmentation, implementing Detectron 2D for 3D volume estimation from 2D contours, and training the system to distinguish real callus from simulated sample to ensure reliable measurement. The workflow involves sterile image acquisition of callus samples, followed by deep learning-based segmentation using the model of machine learning to isolate callus regions. Segmented top and side images are then used for contour-based 3D volume reconstruction. Finally, morphological features such as shape, density, and surface texture are analyzed to generate an objective quality score reflecting the callus regenerative potential. This part of research process and result analysis present the methodology, experiment setup, and data collection process. Key results are analyzed and interpreted, highlighting important findings and trends. Several deep learning models were evaluated for callus segmentation under sterile conditions. First of all, we are going to talk about the UNet, which excels at capturing fine boundaries and segmenting complex shapes. Its encoder and decoder structure allows precise pixel level segmentation. However, it's sensitive to lighting variation and may struggle with irregular texture or shadow. And then we have MaskR-CNN, which provide instant segmentation, distinguish individual object even in the clutter image. It produces detailed mask for each callus, making it ideal for single sample containers. Its main limitation is lower training on large datasets due to the complexity of its region proposal network. And we have the FCN, which is fully convolutional network, which is lightweight and fast, suitable for real-time segmentation or low resource environment. However, it shows lower precision in capturing intricate morphological details, which may reduce volume estimation accuracy for irregular callus shapes. Detectron 2 was chosen as the core framework because it integrates segmentation and volumetric reconstruction, providing a scalable, reproducible, and non-invasive method for estimating callus volume while retaining high precision. Its modular design allows for future expansion to multiple plants, species, or additionally phenotyping traits. Since machine learning requires a large amount of data and bamboo callus tissue is limited, we use mashed potato as simulated material. The volume of each mashed potato sample was accurately measured and used as ground truth for training the machine learning model. The system captured top and side images of callus non-invasively to preserve sterility, then use the deep learning model to segment the callus accurately. Detectron 2 reconstruct 3D volumes from the segmented 2D images, while morphological features like compactness and surface irregularity are extracted to evaluate the quality of the bamboo callus tissue. This metric generates an objective score predicting regenerative potential, creating a non-destructive, reproducible, and high-throughput pipeline for monitoring callus growth over time. After training the model with 10,000 paired samples, we evaluated on 15 on-scene test image. The predicted volumes closely match the real measurement, with most samples achieving over 95% of precision and several exceeding the 99%. These results confirm that the proposed system can provide non-destructive, high precision, and reproducible data, supporting data-driven decision in bamboo tissue culture and laying the groundwork for scalable high-throughput phenotyping. Research conclusion and outlook. Finally, we are going to summarize the main conclusion of the research, and we also propose the duration for future studies. Future work will expand the system to real callus sample for multiple bamboo species to improve cross-species generalization. Quality assessment metrics will be refined using more diverse data to better predict embryogenic potential. The pipeline will be integrated into laboratory workflow with automated image capture, segmentation, volume estimation, and growth curve generation. Additionally, the system will be extended to other plant species, creating a scalable, high-throughput platform for continuous, non-invasive monitoring of callus growth, supporting broader tissue culture and plant biotechnology research. This work presents a non-invasive machine learning system that accurately measures, segments, and evaluates callus growth while preserving the sterility. It reduces manual labor, improves precision, and enables high-throughput monitoring with future expansion to multiple species and refining quality metrics. It lays the foundation for fully automated machine learning-driven plant phenotyping. Thank you very much.
This is interesting topics. I have the question. How many samples have used in your testing training sets?
10,000 per sample. 10,000 top and side pictures to reconstruct 3D volume shapes.
Can test your program with some new colors in other species. I think you can ask for your supervisor for help, and I think this is more convincing.
Yeah, right now the model right now can difference between mashed potato sample and real callus tissues. Right now the system can know the difference between both of them, but right now it's not possible to difference each species of bamboo callus tissue.
No, it's not real callus.
Yeah.
So you need to use the real callus to do your training, your model training.
Yeah. I also use real callus sample. I only use mashed potato because machine learning needs a large amount of data. As I use 10,000 pictures of mashed potato, for if I have to obtain the 10,000 picture of real bamboo callus tissue, maybe it will take much time that I can prepare for mashed potato. The mashed potato was only used to teach with the machine learning to estimate the volume, and then I use the on seeing 15 pictures of real callus tissue. The result that I have is on real callus tissue, but not on mashed potato. Thank you very much.
From use machine learning model.
Yeah.
And how many times you test your sample?
How many times?
Yes, you make it.
Mm. How much time did I train the model? 15 times, 15,000. Yeah, 15,000 times. Yeah. Thank you very much.
Okay, let's welcome the speaker poll. And the title of the speaker poll is in the meeting. The title of speaker poll the other speaker introduction of the generation of the mono factors transparency. There is okay, let's welcome the speaker by she is in the meeting. Okay, the sixth speaker is The speaker six, the title is Directing Superior Digital Columns. We are lobbying results to factory and to profit analysis.
Good.
Good morning, everyone. I am Guo Zheng, and today I'm delighted to present my research title, Developing Superior Dendroclasm Esper via Polyploid from Introduction to Wood Property Analysis. Let's start with the research background. First is acknowledge that the significant value of bamboo. Bamboo is a remarkable plant with multifaceted importance. Ecologically, it plays a crucial role in carbon sequestration, soil conservation, and maintaining biodiversity. Economically, it supports a thriving industry encompassing construction, furniture, papermaking, and edible shoots. And ornamentally, it elegant from grass landscapes worldwide. In China alone, the bamboo industry reached a struggling 415 billion RMB in 2023, highlighting its immense economic potential. However, bamboo breeding faces considerable challenges. One of the primary hurdles is its long and unpredictable flowering cycle. Some species flower only once every several decades or even a century. Compounding this issue, the low seedling rate of bamboo, which severely limits genetic variation. These factors make traditional breeding method inefficient and hinder the development of improved bamboo varieties. Our target species is Dendroclasm esper, a giant climbing bamboo with diverse uses. Its young shoots are edible, offering a nutritious Food source where its study comes are ideal for construction, furniture, and handcrafts. Additionally, its large foliage and impressive size make it a popular choice for landscaping. Polyploid genetic phenomenon where organisms have more than two complete set of chromosomes present a promising solution to bamboo breeding challenges. Polyploid plants typically exhibit desirable traits such as larger organs, enhanced stress resistance, and improved quality. Traits that are highly valuable for bamboo cultivation and utilization. Now let's turn to the research innovation. This study has two key innovations. First, it's the first time to use bamboo colors as an experimental material for polyploid introduction. Colors, a mass of undifferentiated cells, provides a versatile and efficient system for genetic manipulation. Second, we employed two introduction methods, solid media co-culture and liquid suspension immersion. For the first time in bamboo polyploid breeding, the dual method approach allowed us to compare and optimize induction conditions, increasingly the likelihood of successful polyploid generation. The research significance of this work is threefold. First, limited prior research exists on polyploid breeding using bamboo colors as the experimental material. Most previous studies focus on the other plant tissue, leaving a gap in our understanding of color-based polyploid induction in bamboo. Second, this research provides new insights for developing new bamboo varieties and addressing the limitation of bamboo clone propagation. Clone propagation will efficiently lead to genetic uniformity and reduce adaptability. Polyploids introduce genetic variation and improved traits. Third, the study enriches bamboo germplasm resources. These new resources can support policies such as using bamboo as a substitute for plastics, a crucial step towards reaching plastic pollution and the contribution to carbon emission reduction through bamboo's enhanced carbon sequestration capacity. Next, let's outline the research objectives. Our first objective was to establish a stable polyploid breeding system for Dendroclasm asper using embryonic colors. Second, we aim to optimize coqueline-induced polyploid condition, including coqueline concentration, treatment time, and induction method. Coqueline is a widely used chemical agent for inducing polyploid by inhibiting chromosome segregation during cell division. And optimizing its application is key to maximize introduction efficiency while minimizing damage to plant cells. Third, we sought to characterize the morphological, cytological, and anatomical traits of the induced polyploids. Understanding the traits is essential for evaluating the superiority of polyploids plants and their potential applications. Now let's dive into the material and method. For the plant's material, we use embryonic colors derived from Dendroclasm asper, short buds. Colors were cultured under the control condition, a temperature of 25 Celsius and dark culture, which promotes colors growth and maintenance. We employed two introduction methods. For the liquid suspension method, we test the coqueline concentration of 0, 15, and 100 milligram per liter with treatment time of 24, 48, and 72 hours. For the solid media method, concentration ranged from 0 to 5 milligram per liter, and the treatment time of 7, 14, and 30 days. The control group with no coqueline allowed us to assess the effects of coqueline along on color growth and employed introduction. To identify the polyploids, we use a multi-steps approach. Flow cytometry, cytological observation, anatomical analysis, and morphological measurement. Moving to the key result, let's first discuss the optimal induction conditions. For the liquid suspension method, our goal was to minimize the impact on colors. Differentiation will maximize the polyploid induction. We found that a treatment of 100 milligram per liter coqueline for 48 to 72 hours yields the best result. At this concentration, the time range, the induction rate was relatively high and the colors differentiation was not severely inhibited. Additionally, our observed that under the same treatment duration, the browning rate of colors gradually increased with the higher coqueline concentration. Colors browning is a sign of cell damage, so this trend indicates the high coqueline concentration can be toxic to color cells, highlighting the importance of optimizing concentration to balance introduction, efficiency, and cell viability. For the solid media method, the optimal conditions were different. We determined that the coqueline concentration should be below 15 milligram per liter with cultivation period of 14 days. Concentration above 15 milligram per liter led to excessive color browning and reduced survival rates. Interestingly, prolonged treatment under low coqueline concentration had a minimal effect on color health. This suggests that the solid media method is more suitable for low coqueline concentration and moderate treatment times, as the solid matrix may slow the absorption of coqueline, reducing its toxic effects. Next, let's examine the result of polyploid in the identification using flow cytometry. For the liquid suspension method, we regenerated 44 plants, of which 13 were polyploids, representing a significant induction success rate. And for the solid media method, 3 plants were regenerated, with 1 being a polyploid. The flow cytometry graphs clearly showed distinct peaks corresponding to different ploid levels. The 6-ploid control group had a peak around 116. The 12-ploid groups around 320, and the 18-ploid group around 418. Those results confirm the successful induction of ploids with varying germplasm seeds. Now let's explore the polyploid traits, starting with morphological measurement. We observed several notable traits. First, leaf thinning. Polyploid plants have significant thicker leaves compared to diploids significantly. Specifically, the leaf thickness of 12-ploid plants increased by 47% compared to 6-ploid plants. Second, increased leaf size. Comparisons of leaf length width and length to width radial revealed that the 12-ploid plants showed a trend towards larger leaves than 6-ploid plants. Larger leaves can enhance the photosynthetic capacity, improving the plant's growth and productivity. Third, enlarged stomata are crucial for gas exchange and transpiration. The guard cell of 12 ploid plants are 14% longer and 19% wider than those of 6 ploid plants. Larger stomata may improve water and gas exchange efficiency, contribution to better stress tolerance. Next, we evaluated the 3 major components of wood: cellulose, lignin, and hemicellulose. For lignin content, we found that the 12 ploid plants had significantly higher lignin content in all tissue compared to both 6 ploid and 18 ploid plants. Lignin provides niche structure, support, and rigidity to plants, making high lignin bamboo ideal for construction and structure application. For the cellulose content, the 12 ploid plants also had significantly greater cellulose content in all tissue than 6 ploid plants. Cellulose is the main component of plant cell walls, and its response to tensile strength. Higher cellulose content improves bamboo's durability and suitability for fiber-based products. For hemicellulose content, the trend was more varied. In leaves, the hemicellulose content of 12 polyploid plants was significantly higher than that of 6 ploid plants. However, in main branches, the opposite trend was observed. Hemicellulose plays a role in cell wall structure and water retention, and this tissue-specific difference may reflect adaptation to different functional requirements. Looking ahead, we have several plans for future works. First, stress resistance validation. We will adjust the 12 polyplant to drought and cold stress treatment to evaluate their drought and cold resistance. This will help determine their adaptability to different environment conditions and expand their cultivation range. Second, material property evaluation. We will measure the mechanical properties of 12 ploid plants, including bending strength, compressive strength, and hardness, and correlate this with their chemical compensation date. This will provide a comprehensive understanding of how polyploid affects bamboo's mechanical performance, guiding its application in varied industries. Third, cultivation scale up. We will promote vegetative propagation techniques such as diversion and tissue culture for polyploid plants to increase seedling production. This will facilitate the large-scale cultivation and commercialization of superior polyploid bamboo varieties. Now let's move to conclusion. Based on our result, we established the optimal induction protocol for dendroclamsum asper polyploids. For liquid suspension method, treating colors with 100 mg chalkline for 48 hours is recommended. The polyploid potential of dendroclamsum asper is evident. The 12 ploid plants showed significant improvements in key traits compared to 6 plants. The 12 ploid plants showed increased guard cell size, leaf thickness, and leaf size compared to the 6 ploid plants, suggesting improved energy use and stress tolerance. In terms of material properties, the 12 ploid plants had higher lignin and cellulose content than 6 ploid plants, indicate potential for construction and structural uses. In summary, this research established a reliable polyploid breeding system for dendroclamsum asper, identifies optimal induction conditions, and characterize the superior traits of polyploids. The funding of this study not only contribute to the advancement of bamboo breeding technique, but also provide valuable germplasm resources for the bamboo industry, supporting sustainable development and environmental protection. Thank you for your attention. I will now be happy to answer questions if you may have.
Why choose the use of station and this will create the quality, but not the solution video proportion.
Because under the test we get generated 14 reborn plants by liquid one, but on the solid one we only get 3 plants. So we may think the solid one may cause more damage on color cells, but the liquid one with short culture time, so it's the damage to color cell may less.
Presentation has welcome of the report, the round from 10. It's her title, that is the economics, teachers interaction and the generation of Zimbabwe London campus.
Everyone. My name is Wang Tiantian. I'm a first graduate student from Bamboo Industry Institute, Zhejiang A&F University. My presentation today is about callus induction and regeneration from immature embryos of Kimono bambusa pinchanensis. My presentation is divided into 4 parts: background, content, results and analysis, and conclusion. First, let me introduce Kimono bambusa pinchanensis. It is a species within the genus Kimono bambusa of the subfamily Bambusideus. It's characterized by its 3 branch carps and sympodial-monopodial mixed rhizome system. This species resembles C.H. quadrangulus, but differs in having longer area roots at the nose. As a high-altitude bamboo species, it produces shoots in autumn, which is rare among the edible bamboo species. Its shoots are thick, tender, crisp, and sweet, benefiting from the pristine air, soil, and water in mountainous regions. This makes it one of the most distinctive and excellent shoot-producing bamboos in southwestern China. This figure illustrates flowering stand of Kimono bambusa pinchanensis. This species requires 2 full years to complete its flowering cycle from flower bud to anthysis. Because bamboo species are characterized by long flowering intervals and low seed rates, Cumulobambusa pinchianensis, predominantly distributed at high altitudes of 800 to 1600 meters, has specific cultivation requirements. Conventional propagation techniques, such as seed germination and cutting, are constrained by high consumption of mother plants and low multiplication coefficients, leading to slow breeding progress and insufficient supply to meet market demands. So this study established a callus induction and regeneration system for C. pinchianensis and successfully obtained regenerated plantlets. This work lays a foundation for developing an in vitro propagation system and facilitates future breeding of new varieties using gene editing technologies. Next part is research content. First, screening the embryonic developmental stage. Basal medium and plant growth regulators for callus induction. Second, determine the optimal 2,4-D concentration and ZT 2,4-D combination for callus proliferation. Third, screening different plant growth regulator combinations for callus differentiation. Fourth, identifying suitable plant growth regulators for root induction for callus. And you can see in the right chart, callus proliferation rate increased initially and then decreased with the embryo developmental stage. Stage 2 were identified as the most suitable explants for callus induction. The highest induction rate was observed on MS medium, so MS was selected as the basal medium. This is the process of callus induction. The embryo began to swell after 3 days. Callus initiation was observed between 9 and 12 days, and by day 21, most embryos had produced callus. 2,4-D had a significant effect on callus induction. The highest induction rate was achieved at a concentration of 2.5 mg per liter, while the browning rate increased with higher 2,4-D concentrations. Therefore, the addition of 2.5 mg per liter to 4D is recommended for optimal callus induction. While the addition of either 6BA or NAA significantly reduced the callus induction rate, however, the inclusion of NAA was found to be effective in lowering the browning rate. The treatment with 0.001 mg per liter picogram resulted in the highest callus induction rate and the lowest browning rate. A concentration of 1 mg per liter ABA yielded the maximum callus induction rate of 70%. This is the callus induction and regeneration system. Result 2, callus proliferation. The proliferation coefficient increased and then decreased with the rising of 2,4-D concentration and reaching a maximum of 2.19 at 1 mg per liter 2,4-D, which produced pale yellow, highly granular callus with vigorous growth. This is different types of callus. On the basis of 1 mg per liter 2,4-D, different concentrations of ZT were added. Adding 0.2 mg per liter ZT resulted in a higher callus proliferation coefficient of 2.57 and a lower browning rate. Result 3 is callus differentiation. Based on range analysis, the plant growth regulators on callus differentiation from greatest to least was 6BA greater than NAA greater than KT. The highest differentiation rate of callus was achieved with treatment 4, which contained 1 mg per liter 6BA and 0.5 KT and 0.25 NAA. Based on Table 4, 6BA had a significant effect on callus differentiation at 0.05 level, whereas KT and NAA were not significant. Further analysis of 6BA had revealed that the highest differentiation rate of callus was achieved with 1 mg per liter 6BA, which differed significantly from other concentrations. For rooting, treatment 4 had produced the highest average root number, with roots that were thick, straight, and green. This is tissue cultured plantlets after acclimatization and transplantation. In conclusion, this study established a callus regeneration system for Chimnobambusa pinchanensis using immature embryos as explants. The main conclusions are as follows. Notably, browning was the major regeneration constraint. Experiments indicated that only NAA and picogram partially elevated browning. That's all for my presentation. Thanks for your attention. The reason I choose this bamboo species is because it's an excellent shoot-producing bamboos in southwestern China. And also it produces shoots in autumn, which is rare among the edible bamboo species. So it can produce the large economic value. And second, I think this species can lay a foundation for further gene editing technology research. Thank you. Okay. No more question. Thank you.
Okay, the last one is also from that speaker by, Yang Fan, sorry, the title is silicon reduced packaging of silicon accumulation in multiple countries by rising expression of transporter in the shop.
有份是的，我不知道。
And this is the last one of this half session, and we will wait for the 30 minutes. 30 minutes.
这是线上微信，下次。你加我的微信，我直接写到这里。
Hello, everyone. My name is Yang from Zhejiang Agriculture and Forestry University. Today, the title of my presentation is to select reduce carbon emission in monsoon, where political prediction and down-regulation the methodology for the teams in monsoon. As we all know, carbon is one of the most abundant carbon sources, including plants, as we see here. Under the cadmium stress, cadmium severely inhibits the normal growth of roots and shoots in rice and wheat. And Mosu bamboo is one of the most important bamboo species in China. And Mosu bamboo can not only provide raw materials for industrial uses, but also it shows can be used as a traditional vegetable in our daily life. However, according to surveys, the concentration of cadmium in some Mosu bamboo shoots have exceeded the standards for people to eat. So this phenomenon have led to a potential food safety. And in my previous study, I also found that under the cadmium stress, cadmium severely inhibits the normal growth, as we can see in this picture. The growth of Mosu bamboo root and shoots was significantly inhibited under the cadmium stress. And Mosu bamboo has shown the high capacity for cadmium accumulation, as we can see here. Under the with the cadmium concentration in the Mosu bamboo shoots have exceeded 200 kilogram per kilogram. Then there is a question: how to reduce the cadmium concentration in Mosu bamboo shoots? Some studies have reported that there are three major techniques to reduce cadmium concentration, including the water management, genetic technology, and the application of silicon fertilizer. And in recent years, the application of silicon fertilizer have been reported and reduced cadmium, leader, and manganese accumulation in many plant species, including rice, wheat, and so on. And we found that in my previous study. And also bamboo shows a high capacity for silicon accumulation. So I want to know, can we utilize the high cadmium accumulation in Mosu bamboo by the application of silicon to reduce the cadmium accumulation in Mosu bamboo? And what is the molecular mechanism by which silicon reduces cadmium accumulation in Mosu bamboo? So we did the following experiments to evaluate. Firstly, we perform a short-term silica treatment, as we can see here. The left picture is the root elongation of mossy bamboo under the different concentration of cadmium stress. This picture is the cadmium accumulation in mossy bamboo roots. As we can see, under the different concentration of cadmium stress, cadmium significantly inhibits the mossy bamboo elongation. However, with the silicon treatment, it not promote mossy bamboo root growth, but it rarely reduces the cadmium accumulation in mossy bamboo roots. This result indicates that the short-term silica treatment can reduce cadmium accumulation in mossy bamboo. Then we also observe the cadmium distribution in mossy bamboo, root tip and base root, as we can see here. The gray color represents the cadmium signal, and the blue color represents the autofluorescence signal from signal 1, as we can see here. Without silica treatment, we can observe the cadmium in our cells, including epidermis, and the cell region. However, with the silica treatment, we can only observe the cadmium signal in epidermis region, is here. And the sum result was observed in base root. This result indicates that short-term silica treatment can alter the cadmium distribution in mossy bamboo roots. In previous studies, and some studies have reported that silicon and cadmium can form a complex, and which was isolated in cell 1. Then we observed the cadmium and the silica complexation in the cell 1, as we can see here. This is the cadmium and the silicon complexation. We found that after the silica treatment, more silicon and cadmium complexation was observed in epidermis region, is here. However, with less cadmium and silicon complexation was observed in still region. So this result indicates that after a short-term silica treatment, more silicon and cadmium complexation was isolated in cell 1. This result was also confirmed by detecting cadmium abundance in the epidermis region and the still region. To form this result, we detected the cadmium concentration in the cell 1, as we can see here. After a short-term silica treatment, the cadmium concentration in the cell 1 was also increased in the root tip and base root. However, we also detecting the cadmium concentration in the cell sample. The figure B and figure D shows the cadmium concentration in the cell sample of root tip and base root. After the silica treatment, the cadmium concentration in the cell sample was also reduced. This result indicates that after a short-term silica treatment, more cadmium was isolated in the cell 1, resulting in less cadmium entering the cell sample. Then we also compared with the different components of cadmium and the silicon accumulation in the cell 1, including the actin, hemicycle 1, hemicycle 2, and the cellulose, as we can see here. The hemicycle 1 shows the highest cadmium and silicon accumulation, with the silicon treatment, the cadmium concentration in the humus one was also increased. So this result indicates that humus one is the main enrichment for site for silicon and cadmium accumulation, and the silicon treatment can also reduce cadmium concentration in the humus one. Then, based on above results, we can conclude that after the silicon treatment, silicon and cadmium can form a complex and isolated in CO. Subsequently, the influx of cadmium from steel region to external region to steel region was reduced. In the previous study, we found that silicon reduced cadmium accumulation via physical prediction. How about long-term silicon treatment? Then we perform a long-term silicon treatment to investigate the long-term silicon treatment on the cadmium accumulation in moss bamboo. As we can see, figure A. Without silicon treatment, the growth of moss bamboo root is short, is similar. However, with the cadmium stress, the growth of moss bamboo root was significantly inhibited compared to without cadmium stress. However, with the silicon treatment, it can promote moss bamboo root and shoot growth under the different cadmium concentration. Then we also calculated the cadmium uptake accumulation in root and shoot. This is the cadmium uptake and cadmium accumulation in moss bamboo root and shoot. As we can see here, after a silicon treatment, the cadmium concentration in the moss bamboo root was also reduced. So this result indicates that long-term silicon treatment not only alleviated cadmium toxicity, but also it really reduced cadmium accumulation in moss bamboo. Then to form this result, to avoid the formation of silicon and cadmium complexation in the solution, then we performed a silicon pretreatment assay. That is, say, moss bamboo silicon was treated with or without silicon for 9 days. Then the silicon was transformed to a solution with the same concentration of cadmium, and we found some result. As we can see, after a long-time silicon pretreatment treat, the cadmium uptake and accumulation in moss bamboo root and shoot was also reduced. In the short-term silicon treatment can alter the cadmium accumulation. How about long-term silicon treatment? Then we also observe the cadmium distribution in moss bamboo root after the long-term silicon treatment. As we can see, regardless of whether silicon was application or not, we can observe the cadmium signal, that is the gray fluorescent signal, in all cells, including the epidermis, epidermis, and steel region. And this result was further confirmed by detecting the cadmium concentration in the cell wall. We also found that after a long-time silicon treatment, the cadmium concentration in the cell wall was also reduced. This result indicates that long-term silicon treatment did not alter the cadmium distribution in moss bamboo root, but it can significantly reduce cadmium accumulation in moss bamboo root of silver. And some studies have reported that long-term silicon reduces cadmium accumulation by inhibiting the some transporter genes which involved in fundamental accumulation and fundamental transport. As we can see, in my previous study, we found that the accumulation of silicon in rice shoots can regulate the expression of some genes which may be involved in lead or cadmium accumulation. To investigate the molecule of long-term silicon reduced cadmium accumulation, we perform a transcriptome assay. This is the transcriptomic sequence data. And the number and this family have been reported inverted in cadmium uptake by many months. Then we isolated these genes, and we found that after the silicon treatment, there are 3 genes with down-regulated expression. So I hypothesize that after a long-term silicon treatment, maybe down-regulation the expression of these genes. For example, P5 and the P2. So the down-regulation of these genes maybe reduce the cadmium uptake and accumulation in mouse bamboo. Then to confirm the P5 has the cadmium activity, then we heterologously expressing P5 in the yeast mutant, which is defective in cadmium uptake. As we can see here, without silicon treatment, expressing P5 is similar to that the growth of yeast expressing P5 is similar to that expressing empty water. However, with the cadmium stress, the growth of yeast expressing P5 is significantly inhibited compared to expressing empty water. This result was also confirmed by detecting the cadmium concentration in the yeast cells, as we can see. Expressing P5 also increase the cadmium concentration compared to expressing empty water. So this result indicates that the P5 has the cadmium activity. Then this is to confirm this result, we express this P5 in Arabidopsis mutant, which is defective in cadmium uptake, and we find the same result as we can see this picture. Under the cadmium stress, the growth of plant's root was significantly inhibited when expressing P5 compared to wild type or 18.1 mutant, and we also detected the cadmium concentration in the root and shoot, as we can here. Expressing P5 also increases the cadmium concentration in roots and shoots. So this result indicates that P5 has the cadmium activity. And the same result was further confirmed by expressing this gene in the rice mutant OS95, which is defective in cadmium uptake. So through the 3S, which shows the same result, that is the P number 5 has the cadmium accumulation. Then we also confirm the subcellular localization of P number 5. As we can see here, this is a GFP, the gray color. We express GFP only. We can observe the GFP signal in all cells, including plasma and nuclear. We express GFP field with P number 5. We can only observe the GFP signal in plasma member, which this result indicates that P number 5 is a plasma member transporter, and the sum result was observed by expressing P number 5 in unnew epidermis cells. And we also found that the P number 5 is localized in all cells. So through the above result, we can conclude that regardless short-term or long-term selectivity, post-treatment can also reduce the cadmium uptake and accumulation in most bamboo root and shoot. However, the molecular mechanism by selecting reduce cadmium accumulation between the long-term and short-term selectivity is different. After a short-term selectivity, selection reduces cadmium accumulation while physical fixation cadmium in hemocyte 1 or cell 1. However, after a long-term selectivity, selection reduces cadmium accumulation by down-regulating the expression of genes. For example, the P number 5 and the P number 2 and the P number 5 as a plasma transporter have been confirmed has 2 cadmium activity. So, in the end, I would like to express my gratitude to Professor Shaw, my tutor, for his invaluable guidance throughout all my experiments. And I also thank to every member of Shaw Lab for his assistance and support. That's all for my presentation. Thank you very much.
Thank you for your confident presentation. And if anyone wants to ask questions. I have a question for you. Page 21. In the presence of garlic cost, there's no difference in the growth of its expression in P95 compared to control. Under the part stress, however, in the presence of garlic cost, the growth of its expression P95 is adversely inhibit. Could you explain this question?
Okay, thanks for your question. Okay, as we can see here, in the presence of glucose, the genes cannot induced. However, in the presence of galactose, this expression of genes can be induced. So we can observe that in the presence of glucose, the growth of it expressing P number 5 or empty vector, the growth is similar. However, in the presence of galactose, so this P number 5 can be induced expression. So we can observe that under the different cadmium concentration treatment, expressing P number 5 also shows significantly impaired compared to the empty water. So that's all. Thank you.
Wait. Check the online question. No more question. Okay.
Okay, the of this participants will end. And I will thanks to the sixth speaker for the excellent presentation and for your excellent achievements. And thanks, George. Thanks for your time and thanks everyone, every listener. So this half session is over and we will come back at 10:14 coffee time. So, okay, this is the end of the session.
不是很好，因为那人打都会有一些都打很低分，有的都打很高。
哦，真的。
就是肯定有昨天有些讲到的，如果放在今天来说，他可能就不行了，因为我们可能打挺好的。因为没办法，其实最好是都是一批人来打是最好。
对。
对，这样打到时候我们就考虑。就是不用这个东西。有差别吗？
我是刚才那个。然后刚才那个人刚散的意思。
你不能去带队了，我都过了，但是你都过了，都给我安排。
不能到一点，不能到一点。要不然知道一点是非。
像去年有一个说不公平的原就是那个什么黄花玉因为院长在之后，他说必须按照他给打分的，就是因为打分标准不一致，有的有一些惨败，所有的分数比另外一层都要高，但是他说不能够额外的说要3秒15秒钟做的学生。反正最后的话就是大家不能这样，结果说不定正常的也好，都在一个不通里面，都在尴尬。这是好几个学生可能有错的。对啊，这样也你像我们这个跟污染有4个下水，然后有些下水吧，就是特别高，还有评定不一样的，因为还是才差2个3个学月，这个学的是大量不一样。以后我在发图，基本上在看我好的进度。但是其实我也是的，差不多。这个没有说。
现在问的是我的情很忙，我3个礼拜不是我对，而且跟我说，他一开始我听问题跟我的是那个，然后我脑袋想聪明，我一直跟什么跟风雨这个东西，然后才学习，我说以前还说了嘛，然后这个问题啊，觉得都那个感觉做结束，但是我真的不让他在那边搞事，真的没有知道有，我就一直没有做，那是什么呀？啊，没有别的，他不需要什么也可以进去，我啥也没接着来。真没看他什么一点，完全的。这个他看不成不想。第二条。我不知道。我怎么说？
郭志是你的主管。对，他说讲哎呀，他做的是那个，对，后边还有什么？还有啥学？好像。看他来做。先会出去。 哦，还有这个是领导公司，这个按照这个规定。啊，然后这是下一个小手。对，这个标准也按照标准。不过测那个力学定律，看材料。
我就在这里，那我撒线，没事儿，他说那不一样，不一样。它是评委，还有一个 model， 那个水稻，你知道吗？要不，我已经 OK， 这我基本上有您的那个名字牌吗？没有，就这样就行。这里面有的。你去找，应该没有吧？没事儿，这样子。没关系，没关系。但是他这个设计，就是有灯的，
那我们是不是那个会？
这就结束了。
我请假。在学生很多时间这个假。
接着出来
这401当然有错误概念出来，那这个业余水的。我觉得
那没大9人对吧？那个4个人，报告是1天。哦，然后很慢。
因为我们说。全体这个方面，
就不要有最初一点的，但是做直接去开。我打算去做这个我在四川女战时候又经常挑战。我就太了解女人也不太认识。我们3两点都然后那里面他的这个在这个组，但是我觉得很好，像两个人上来是说还是但是看的放完有些人不知道，然后那个不是他们都突然死神，我本来想我当时因为上次评委从头跳到一个然后希望
我也不懂他们的分级，我偏材料。
那你做他说那个
会做，但是我偏纤维素，因为
昨天下午我们在去一个
对塑料的那个。
这样，就是一般是就是我一开始我以为这个是2个都在一起，但是吧，其实现在今天的话是那个第4类型，第1个和第3个那个分会场，好吧？这分会场那你就在那个分会场里接受了，这类是第1个，我以为是还是那个后这个是我已经联系好，我他们说的。
我也感觉真正后面怎么样和那些系统上不谈的，还有点关系。你是要主要是哪个不能？
因为我们
因为它力决定的，竹片的力决定的，还是纤维素含量，还是纤维的性质？ 那些一句话出事儿，你长成了成熟的2到3期，按照市场附加2到3年嘛，人都会让消费者12到15的。还有一个当然，这个都可以，目前使用的是纯黄的，不去年咱现在就工业上空间水平就高。咱学校地址有大几就有，我们那个从制度
所以跟我们刚才提的内容的是
车里的大就是做一个装饰在里边，那个外面的那个还不会用他现在目前做的可以让真的现在目前车上我了解的情况，它外边还是用但里边的装饰地板啊那个东西，或者说有些塑料，比如说像咱墙上的那些塑料，它里面有可能会加一些纤维，就是轻质化。王宇说：“我其实用先进纤维高才用。”包括对有别的是这样的，这样的是网络化的一个管理款。一般是对，都是把谈到您的旧的要说的话，它对路牌的性质不只讲究，但是每个这种路牌的这个性质上，就是如果放出来它干掉这个位置的话就造成应该是长的地方类似的想法是我们主要就是关注压缩进8倍。对对对。所以像我们这样就是我们主要是让先子弹但是你看不出来，那么所以这小带来什么伤？所以这个问题啊，你出来，然后或者说是看这个也可以以前或者这个就是做哪个感觉，包括造纸，包括要求这个以前他就基于是不一样的评价评价。对，包括我们的对，希望没有。
Okay, welcome back. I'm the moderator of this session. My name is Golden, and I'm teacher of our institute. So maybe I don't need to introduce myself, but I'm most of you maybe 1994 in our building. So to save time, let's continue and welcome the first speaker in this session, 叶小丽。Her title is the traditional and naturalistic characterization of DEK2 in living biosynthesis in North Bamboo. That's better.
Good morning, everyone. I'm Ye Xiaoli, come from Zhejiang Agricultural and Forest University. And the title of my presentation is Functional and Mechanistic Study of CAT2 in Lignite Biosynthesis in Moso Bamboo. I will elaborate on this study from 3 parts, starting with the research background. Moso bamboo is the most widely distributed bamboo species in China. It's characterized by fast growth, high linear cellulose, excellent mechanical properties, and low ash content. Making it an ideal role in paper making, furniture, manufacturing, construction, industrial production, and other fields. The chemical composition of Moso bamboo mainly consists of lignin, hemicellular, and cellulose. The lignin is a complex phenolic polymer that is typically deposited in the cell walls and closely cross-linked with substance, enhance the strength and toughness of the cell walls and playing a crucial role in plants' growth and development. Lignite biosynthesis is a complex and branched process that involves the participation of multiple enzymes. Among them, CAT is the final enzyme in the synthesis of lignite monomer. And its primary function is to catalyze the reduction of 3 types of cinnamaldehydes into their corresponding cinnamol alcohol. At present, in-depth studies on CAT have been conducted in various other species, yet research on CAT in Moso bamboo remains relatively scarce. Therefore, this study focuses on CAT gene in Moso bamboo, aiming to provide a theoretical basis for future research on lignite biosynthesis and offer a theoretical support for its potential application as a high-quality biomass energy source. Firstly, we identified 14 CAT genes in Moso bamboo. Drawing on the research finding of Professor Wei Qiang, this study analyzed the expression level of CAT gene in the 18 internal of Moso bamboo during the rapid high growth period. Based on the expression model, we roughly divide the sample into 3 regions: the cell division region, the cell elongation stage, and lignite accumulation stage. During the rapid high growth of the 18 internals, 5 genes exhibited relatively high expression levels. Specifically, the expression level of CAT6A12 continually increased in the cell division and cell elongation region, but significantly decreased in the lignin accumulation region. In contrast, the expression level of CAT1 and 2 display a gradual upward trend, and CAT2 maintained high expression throughout the lignin accumulation region. Relevant studies have reported that enzymes involved in the lignin biosynthesis pathways form homodimer and heterodimer, which interact with each other for metabolic channels. Therefore, to investigate whether members of the cat family in moso bamboo can form dimer to enhance enzymatic reaction rate, we selected cat 1, 2, and 8 with relatively high expression level for experiment. The results show that cat 1, 2, and 8 enzymes only form homodimer in its cells, whereas they interact with each other and are capable for forming heterodimer in plant. For further in-depth research, we collected relevant samples in Banqiao Town, Lin'an District. We sampled the 18 internal of young moso bamboo plant at height of 3 meters, 4 meters, and 6 meters, with corresponding internal lengths of around 4 centimeters and 13 and 31 centimeters. These samples correspond to the cell division stage, cell elongation state, and cell lignification state. Next, we determine the lignin content of these 3 representative internal. This result showed that the lignin content of the 3-meter tall young bamboo was about 28 milligram per gram, and the 6-meter tall young bamboo was about 15 milligram per gram. The determination of CAT enzyme activity in these 3 representative internals revealed that the variation trend of CAT enzyme activity was consistent with lignin accumulation, increasing with the growth of young bamboo. The enzyme activity has been significantly improved. We also performed a Pearson correlation analysis between the lignin content data and CAT enzyme activity data. The results showed that a positive correlation between the lignin content and CAT enzyme activity. This finding indicates that lignin deposition in bamboo mainly occurs after the cell elongation during rapid high growth, and CAT enzymes may play a vital role in lignin synthesis during the growth of young bamboo. We also detected the expression level of several CAT genes. The results showed that in the 6-meter tall young bamboo, the expression level of CAT2 was upregulated, with particularly high expression in the internal. In contrast, the expression level of other 4 genes showed no obvious increase, and there were no significant difference in their expression level across different parts of the internal. And we propose that CAT2 is likely the primary gene in moso bamboo involved in biosynthesis during the growth of young bamboo plants. To further investigate the biological function of CAT2, we construct an overexpression vector and conducted the rice transformation experiment. Observation and statistical analysis showed that the average plant height of 3 overexpression rice lines was higher than wild types, and their stem diameter was also slightly thicker than wild types. Determination of the main cell wall components in the transgene rice revealed that the lignin and cellular contents were higher than wild types. while the hemicellulose contents was significantly lower. Further detection of the expression level of gene involved in the lignin biosynthesis pathways. The results show that the expression level of lignin synthesis-related genes, including COMT, F5H, and ABCG, were upregulated in the overexpression lines compared with the wild type. Our experimental results can initially indicate that overexpression of CAT2 promotes the synthesis and accumulation of lignins in rice. This result confirms that CAT2 is the key gene in the moso bamboo. Next, to explore the potential molecular, regulatory, and mechanistic of CAT2 in moso bamboo, we use transcriptomics data to screen out some transcription factors that may have regulatory relationships with CAT2, and conducted IS1 hybrid and lucifer assays. To further investigate the binding size of these transcription factors in details, we divided the CAF2 promoter region into four segments and do it separately. The results show that these three transcription factors can bind to the promoter region of CAT2. And partial results from the lucifer assays can also support this conclusion. And the results indicate that three transcription factors may be involved in the lignin biosynthesis of moso bamboo by regulatory of CAT2. Through experiments such as lignin content determination, CAT enzyme activity assay, and CAT gene expression level detection, and so on, our study confirms that CAT enzyme play an important role in lignification process during bamboo growth, and CAT2 is likely the primary gene in moso bamboo involved in lignin synthesis. And my presentation ends here, and I would like to thank Bamboo Genetics and Gene Plasmid Innovation Team. Thank you all.
Mentioned that CAT is SCG, and so you have entered the gene imaging everything activity.
I already conducted some research on this, and CAT2 showed an in viral enzyme activities, but I'm still working on the substrate preference because cats have three type of cinnamaldehyde. So I'm still working on it, and I didn't present the result.
So this project is about temperature analysis of the roots is in three main and blue natural characteristics. So speaking the Okay. So the topic is about the impact of the different irrigation modes on the growth, yield, and the quality of the cannabis brand listing. That's all I'm saying.
Good morning, I'm the presenter Li Qian. The topic of my presentation today is the effects of different irrigation rates on the growth, total yield, and the quantity of dendrocolizing practice. Due to its outstanding quantities, it has been introduced to Sanya in Hainan. However, research on its optimal irrigation regime in Hainan remains limited. Therefore, we conducted targeted irrigation trials to investigate how different irrigation rates affect its growth, yield, and quality. We employed a randomized block design, 3 composts per plot, with 4 replicates per treatment. PVC barriers enclosed each plot to prevent lateral water movement. 5 irrigation levels were triggered by soil water potential cereals. Moving to leaf physiology, we observed that irrigation regimes differently influenced nutrient dynamics and photosynthetic performance. Crucially, W4 consistently supported higher accumulation of key nutrients, nitrogen, phosphorus, and potassium, which collectively contribute to enhanced photosynthetic capacity. While chlorophyll and certain photosynthetic parameters peaked under another treatments and different stage, W4 exhibited the most stable and integrated performance, effectively balancing nutrient uptake with photosynthetic effectively throughout the growth period. Examining plant stress physiology, the antioxidant system relieved clear treatment effects. W4 consistently exhibited to the loss activity across SOD, POD, and CAT. Azamils allows with minimal MDA accumulation, a direct indicator of memory integrity. This coherent biochemical profile demonstrates that W4 provide irrigation conditions that minimize oxidative damage, thereby preventing circular homestays and redirecting energy towards growth and production. Turning to the root system, we assess both activity and architecture. Treatment W4 results in the highest root activity, indicative of superior physiology function. In terms of structure, W4 developed the most expansive root length and surface area, is strongly associated with soil exploration and nutrient uptake efficiency, while W5 produce a greater total root volume, the specific morphological advantage of W4 likely contributed to its enhanced overall plant performance. For both yield and quantity, W4 performed the best. It achieved the highest short yield along with superior nutritional content, including more free amino acids, soluble sugars, and key minerals. This demonstrates that W4 effective combines higher productive with enhanced nutrition quality. Collection analyzed revealed that bomb short yield was positive correlated with free amino acids, soluble sugar, protein, and phosphorus. Most strongly with free amino acids, lift nitrogen, phosphorus, and potassium were positively correlated with photosynthetic transpiration and root indicator, which in turn were close linked to yield formation. In summer, by enriching when soil water potential drops below -20 kPa, what we call the W4 treatment, we improve the plant's nutrient balance, both the photosynthesis and root function, and reduce physiology stress. Together, this response enable W4 to produce the highest yield while also delivering better nutrition quality in the shoots. Therefore, under the conditions of this study, W4 represents the most effective irrigation practice of the foregoing dichlorocolumnous varieties with both productive and quantity in mind. Thank you for your attention.
Thank you for the tip. Is there any questions for comment? Yeah, so thank you for especially regarding about having for treatment performing that at my last completely pick up how might this relation possible be projected into my regions with different slope types or plant conditions.
It was optimistic for our local standing limb and climate. For another conscience, we recommend using and adjust the their shoulder accordingly.
Okay. And as well in the sticker 2, Chen Zhenli is talking about the community capacity of the rules is extremely influence. So,
Good morning, everyone. I'm Chen Zhenlin. The title of my presentation is Girls Trait Variation Among Three Varieties of Ley Bamboo. At the global scale, limiting human activity and changes in species habitats have jointly driven rapid trait changes in plant population, resulting in pronounced interspecific variation. Climatic and soil variables have been demonstrated to be significant factors contributing to plant trait variation. Bamboo plants are widely distributed globally, and China harbors the world's most extensive distribution and the richest diversity of bamboo species. Their broad geographic regions has led to the emergence of numerous interspecific variants. Ley bamboo, as an economically important bamboo species, has developed numerous variants. Surrounding artificial conservation coupled with natural evolutionary processes. This study aims to analyze growth trait variations among three ley bamboo varieties, integrated with climatic and soil environment data to identify key growth traits and the climatic environment factors driving their variation, as well as to quantify the contribution of each environment factor, with the global goal of promoting sustainable development and conservation of bamboo resources. The key scientific questions to be addressed in this study are: what are the key growth-related variable traits among these three varieties? What are the key environmental drivers of the variation in climatic growth traits? What are the relative contribution of each environment factor? Research content and plans. Analyze the divergence characteristic of phenotypic and physiological traits among the three ley bamboo varieties and predict their key growth trait and their correct environment factors driving the variation. Select a natural distribution area experiment site under different environments. 枣园 bamboo from 德清, 蟹雷 bamboo from 临安, 安徽雷 bamboo from 宁国. Select introduction experiment sites. Under the sun environment, 3 simple plots from 枣园 bamboo, 蟹雷 bamboo, and 安徽雷 bamboo within the 翠竹园 of Zhejiang Agriculture and Forestry University. Sampling was conducted in July and October 2024. Has a standing count of 1 and 3-year-old bamboos were collected from each plot. Parameters to be measured: index growth, phenotypic traits, growth-related physiological traits, and cell physicochemical properties. All three natural distribution sites are located in the subtropic monsoon climate zone, among which the Ningguo area exhibited relatively inferior soil conditions with comparatively nutrient impoverished characteristics. Phenotypic trait analysis revealed that the Anhui ley bamboo from Ningguo region exhibit significant superiority over the other two locations. Comparative analysis of photosynthetic composite across natural distribution areas demonstrate that Anhui lei bamboo from the Ningbo region generally exhibit high levels. Comparative analysis of chlorophyll content and non-structural carbohydrate content across region revealed that Zaoyuan bamboo from the Deqing region exhibit relatively low levels of these components. Analysis of nutrient introduction in leaves and cones of lei bamboo across regions indicated work performance in the Anhui lei bamboo from the Ningbo region. Comparative analysis of nutrient content in roots and resins indicated that the Ningbo region generally exhibit low levels compared to other regions. The following section outlines the bicycle environment profile of the introduction site, where the environmental conditions across cultivated types were closely aligned. Comparative analysis of phenotypic traits at the introduction site revealed the Anhui lei bamboo maintains significantly superiority phenotypic performance relative to other cultivated varieties. Under uniform environment conditions, comparative analysis demonstrated that Anhui lei bamboo exhibit superior photosynthetic capacity relative to other cultivated types. Comparative analysis of chlorophyll content and non-structural carbohydrate levels among different cultivated types revealed that Anhui lei bamboo consistently exhibit higher values in these properties. In the comparison of leaf nutrient content, Anhui lei bamboo exhibit low concentrations, where the crown nutrient content showed similar levels across all cultivated types. Compared analysis of nutrient content among different cultivated types of lei bamboo at the introduction site revealed that intercultivar difference were narrowed through Anhui lei bamboo consistently exhibited low nutrient levels compared to other cultivated types. Metastatic integrating growth trait and environment data revealed significant correlation between leaf phenotypic, chemical, and pigment traits and all environment factors exhibit cell bulk density, Paston, a new max temperature, frost field period, and a new precipitation. Subsequently, a meta-test was conducted between the environment factor showing correlation with leaf phenotypic, chemical, and pigment traits, and the introductors within this group. The result indicated that the leaf area exhibited the strongest interaction with environmental factors. Given the varying degrees of correlation between growth traits and environmental factors, ranging solely on This information to assess key growth traits may compromise the variety of the results. Principal component analysis PCA was deployed to comprehensively evaluate the 8 growth traits. The first 2 principal components extracted by PCA acted for 83.74% of the cumulative contribution rate experienced that measured of the variability in the phenotypic, chemic, and pigment traits. Besides on PCA result, the factors saw conference of the 2 principal components were calculated using standardized data from 8 indicators of the phenotypic, chemic, and pigment traits as independent variable. The results indicated that within the natural distribution area, the Anhui bamboo from the Ningbo region achieved the highest comprehensive score, was the Zaoyuan bamboo from the Deqing region exhibited the lowest score. By integrating Mendes test and PCA, leaf area was selected as the target variable for predicting the importance of environmental factors. The prediction model demonstrated that among the microscale soil environmental factors, available phosphorus, total parasite, pH, and field capacity, along with the macro scale environmental factor mean annual temperature, aeration, and mean annual mean temperature, exhibited significantly greater importance in the model compared to other environmental factors, indicating their correct influence on leaf area variation. In contrast, soil nutrient factors such as total nitrogen, total phosphorus, magnesium, and soil organic carbon showed relatively low importance. To verify whether the key environmental factors identified by the rainforest model in the natural distribution areas, they are significantly influence growth rate when macroenvironmental variation are eliminated. A linear mix factors model was established using introduction site data. The result showed that the contribution rate of environmental factors from the descending order, magnesium, total phosphorus, feed composite, available phosphorus, total nitrogen. The remaining environmental factors exhibit relatively low importance. Among those, magnesium was the most significant positive contributing factor affecting leaf area followed by soil total nitrogen and total parasite. In contrast, soil total phosphorus was the most correct negative contributing factor succeed by field capacity and available phosphates. This is my conclusion. 安徽雷本部 exhibit superior performance in phenotypic trait as well as leaf chemical and pigment properties compared to other cultivated varieties, yet demonstrate relatively weak nutrient accumulation across tissue. Leaf area was identified as the key variable. First trait among cultivars was available phosphates, total parasite, pH, field capacity, mean annual temperature, elevation, and mean annual mean temperature being the character environment drivers of this variation. However, at the introduction side, the importance of magnesium, total phosphates, and total nitrogen increased, whereas the significance of soil available phosphates, total parasite, pH, and field capacity decreased. Thank you for your attention.
How about the three types of bamboo planting?
Thank you for your question. Because preliminary investigation revealed that only that three lay bamboo varieties are cultivated on measurable scale within both natural distribution areas and induction sites.
Well, example of in young parents subjected to usual treatment.
Thank you for your question. Yes, all simple products maintained without fertilization and nutrient treatments.
And his topic is PEPCP4, populated with special activity PEP to expression must have been necessary.
Good morning, everyone. I'm Zhou Jiaqi. My title is Transit Branched 1 Chloride PCF protein P TCP force positivity regulate lateral by the development by activating P2A expression in follow sticks ideals. Most bamboo is not only an ecologically important plant, but also a vital economic resource. Its shoots are nutrient-rich, low in fat and calories, and globally consumed with annual fresh bamboo shoot consumption reaching 2 million tons, 30 to 40% of which are most bamboo shoots. However, a critical bottleneck persists. Under natural conditions, only 5 to 8% of lateral beds are underground rhizomes developing into edible shoots. This extremely low conservation rate severely limits bamboo shoot yield and sustainable development of the bamboo. The TCP transcription factor family is plant specific and plays essential roles in numerous biological process, including branching. Here, we identified a pair of genes and in most bamboo, TCP4A and B, and analyzed their expression patterns during lateral bed development. Response to external seeking and side biological functions and downstream target genes. Before diving into TCP genus, we first validate the role of CK in most bamboo lateral by the development. We conducted external homogeneous treatment experiment using 6BA synthetic CK and two concentrations 50 μm and 100 μm. For the control group, we sprayed the seedings with water. We then monitored the lateral by the outgrowth every 10 days, starting from the 30 day after treatment. The results were striking 50 days post-treatment. The percentage of lateral by the outgrowth in the 6BA treated groups was approximately twice that of the country. Even 60 days later, the 50 mils 6BA group still showed a significant promotion effect with a bad outgrowth rate 1.4 times higher than the control. This data confirm that CK is a potent inductor of most bamboo lateral by the outgrowth. Laying a critical foundation for our subsequent exploration of the molecular pathway linking CK2 by the development. GMOID analyzed revealed 38 TCP family members in most bamboo, classified into class 1 and class 2. with class 2 further divided into CYC, TB1, and CI subfamilies. Consistent with the other plant species, PTCB A and B belong to the CI subfamily. A group less studied for by the development to confirm their function as transcription factors. We performed subcellular localization assess fusing TCP4A and B with GPF and transforming protoplast showed exclusive nuclear localization. Define feature of transcription factors and enables promote binding and gene regulation. We then analyzed the TCP4's expression patterns to link them to literal by the development. Tissue specific qRT-PCR showed both genes are expressed in roots, stems, leaves, and literal buds, with Gustin highlighting stronger activity in rosette leaf buds and seeding pieces. Hunting and specialized role in bud growth. We further examine the expression and key developmental stage LB1 to 4 for above growth bias and S1 to 3 for underground bias. qRT-PCR results show the PTCP4 and B expression gradually increased with by the development. In underground bias, S3 expression was 1.9 and 2.7 times higher than S1. In situ PCR confirmed high expression in literal bias, with intensity increasing as the bias method demonstrating strong positive correlation between PTCP4 expression and literal by the development. To verify their biological function, we generate transgenic biopsies over expressing PTCP4A and B. We obtained twice PTCP4A over expressing less and 10 PTCP-B OE less. All share consistent for types rosette leaf branch number was 1.7 times higher than white tip, with significantly long branch detailed analysis of 6 slides reviewed almost all result leaf best in OE plants development into multiple branches where most WT best remained underdeveloped, providing direct genetic in device that PTCP4A and B are positive regulators of lateral bed development. Research have done that the coil by the element for TCP transcription factors in factors is GNCC motif and oxygen flow transport mediated by P proteins inhibits lateral bed growth. We hypothesize PTCP4 regulate pin genes to modulate oxygen distribution. First, we examine the Arabidopsis genus in OELS and found AP2 and AP7 were significantly upregulated with GGCC motifs in their promoters, extending to moss bamboo. Follow genetic analysis of pin genes from Arabidopsis rice and moss bamboo led us to select P2A and P3B, both with GGNCC motifs in their promoters, making them potential direct targets. We verified the direct binding and activation by East one hybrid and dual luciferase assays. YH assays showed PTCP4A and B specifically bind the P2A promoter fragment containing the GGNCC motif, but not P3B or other P promoters, confirming binding specifically. Dual luciferase seen in tropical living showed transforming P2A like with TCP4A increased the lac rate by 15.6%, while TCP4B increased it by 139%, co-expressing both genus denote enhance activation further than TCP4B, suggesting functional reductance. This result confirm TCP4 directly by the and activate P2A. We test the response to environmental signals, thick and shadow. Negative Q4 by the growth CK treatment repeatedly upregulated TCP4 and B and P2A in contract shadow suppressed TCP4A and B and P2A. This result show TCP4 and P2A integrate external cues to regulate lateral by the development. CK increased the lateral by the outer growth and upregulated the expression of TCP4 and P2A genus. TCP4 transcription factors promote lateral by the development through and list two mechanisms by directly activating the expression of P2A and to modulate oxygen distribution in lateral base and regulate their development. By regulating other transcription factors or oxygen related genus involved in lateral organ development. Okay, thank you for your attention.
Thank you. So, please.
Thank you for your presentation, and I have a question. Do CK and shade have antagonistic effects on the TCP4S and the P2A aspect?
Thank you for your question. Yes, CK induced their expression to promote lateral bias, where shadow suppress them likely will harmony signal crosstalk. Okay.
I have a question. And in your results showing it 6PA treatment, right? 6PA treatment for also bamboo sibling. How many siblings you use this treatment? How many siblings? Can you show me the page of that result? The page of the 6PA treatment result. I think the treatment in the most bamboo. I think. Yes. Remember, okay. Still before it. Still before it. Before it. Still. I think it's in the start. They had introduced the background. Why we should take it. Let's speak Chinese, okay? 但你那个处理毛图的那个片子3道菜，你有做吗？有做吗？
处理猫这
有
个
没有处理？在前面，有处理在前面第3页。OK，这是你做的吗？
这是我师姐做的。
So how many treatments? How many splitting do you use? 多少苗？你用了多少苗在这个处理中 ？How many repeats? 多少声学处理？Because in your picture you only show 3 splitings in 1 point. I think it's not enough. 你处理共同处理了3颗苗吗？
不是。
多少？Because you know different cities, they have the biology difference. 生具有生物差异。So if you only use 3 cities in your treatment and you decide you have this result, I think it's not enough to give this. Can I understand this? 我是说你的生物重复用的有点少，你只有3颗苗。如果你用的多的话，你的这个结果用 A 图应该展示更多的，要不然就没有幸福一样，没有说服。因为不同的苗它的生学差异是很大的，你不能说只处理3颗苗，然后这3颗苗都长侧芽，就说它有作用。
这个是挑出来的，就是作为那个表型对比的。
对，我知道，其实你应该展示。如果你不这样做，因为你没有回答你用多少苗去做这个准备。如果你的苗有很多的话，你应该这个图片应该重新拍一下，不然你去投稿的时候，这个信服度是可能会受到质疑的，因为你只有3个生物学属谱，然后你肯定也种过这个毛图的种子，毛图这个苗的生长差异是很大的。
对，
有的苗你可能不处理它自身就会长侧芽，那你怎么说你这个侧芽是不是处理导致的，还是它本身的生物学差异的，就要把这个生物学的重复的量提上去才能解决这个问题。明白吧？
明白。
OK， 还有 another question。
OK，thank you。
没问题。那就直接就说这么了，就是你这个4A 和4B，你好像做下来它没有任何的生物学功能的区别。就是你做的这个基因染是你做的吗？这个叫什么 TCP 4A 和4B 吗？
对 ，A 和 B。
我给你做下来，从功能到机制都没有任何区别，就完全一样，是吗？那我就问一个简单的问题吧，就是这2个蛋白之间有没有错的？它们能形成复合体吗？有没有可能就是共同对下游之间的调控作用可能会增强？反正你没做过也没关系，就是给你提供一个思路吧，就是以后可以，因为我看你做的已经很完整了，没有投稿吧？
这个已经发出去了。
已经是了。
对，是我师姐的。
哦，thank you. Thank you for your presentation. And let's welcome the next speaker, Wang Shi. Okay. The title is Expression, Protein Interaction, and the Biology Function Analysis, PhD-X1G, in multiple group. Let's welcome.
Hello, everyone. It is my great honor to share our team's latest findings on a topic of both scientific intrigue and practical significance, the flowering regulation mechanism in mossel bamboo. My presentation titled Expression, Protein Interaction, and Biological Function Analysis of PHE-FT1 gene in mossel bamboo. Let me begin by outlining why this research matters. Mossel bamboo is an economical and ecological tree species in China. It exhibits a unique and costly reproductive trait, synchronous flowering, followed by widespread mortality. This phenomenon leads to ecological disruption and significant economic loss, making the understanding of its flowering mechanism a pressing issue. In model plants like Arabidopsis and rice, the FT and HD3A acts as a key component of flowering gene. The mobile single set induces flowering. In mossel bamboo, 16 FT-like genes have been identified, while PHE-FT12A has been linked to flowering regulation. The role of PHE-FT1 remained completely unknown. Therefore, we investigated its expression, protein interactions, and biological functions. We started at the molecular level by cloning and sequencing PHE-FT1. The gene has an open reading frame of 537 BP length, encoding a protein of 178 amino acids, structured with 4 exons and 3 introns. Importantly, sequence analysis revealed that PHE-FT1 contains conserved motifs characteristic of functional FT proteins, including DPD-XP and GXHR. These motifs are known to be essential for PC binding and interaction with FT-like transcription factors, respectively. Next, using qRT-PCR and transcriptome data, we analyzed its expression across tissues and under different light conditions. We found that PHE-FT1 is highly expressed in lateral buds and stems, but low in leaves. Interestingly, its expression was significantly upregulated in inflorescences, with late-stage inflorescences showing 6.8-fold higher expression than early-stage ones. This suggests a role in lateral floral development. Under long-day conditions, PHE-FT1 expression exhibited a clear circadian rhythm, which was absent under short-day conditions. To understanding where PHE-FT1 functions within the cell, we performed subcellular localization assays by fusing PHE-FT1 to a fluorescent protein and expressing it in plant cells. We observed that PHE-FT1 localizes to both the nucleus and cytoplasm, with stronger accumulation in nucleus. This nuclear enrichment is consistent with the non-function of proteins, which often acts in the nucleus as part of transcriptional complexes to active flowering-related genes. To directly test its function, we generated transgenic Arabidopsis plants overexpressing the PHE-FT1. Remarkably, these plants flowered significantly earlier than wild-type controls. Even more convincingly, PHE-FT1 overexpression fully rescued the late flowering phenotype of the FT mutant. These results provide clear functional evidence that PHE-FT1 promotes flowering, much like its FT counterparts of other species. But the story did not end with flowering. We noticed that PHEFT1 overexpressing plants were not only early flowering, but also significantly shorter than wild-type plants. This reduction in height was also observed in the rescued FT mutant background. This indicates that PHEFT1 is involved not only in flowering time, but also in regulating plant height, suggesting a broader role in growth architecture. Similarly, we examined stem morphology and found that stem thickness was reduced in overexpression plants, and this phenotype was also rescued in the FT mutant. Together with the height data, these findings suggest that PHEFT1 influences multiple axes of plant morphology, possibly coordinating reproductive transcription with overgrowth form. A trait set may be particularly relevant in mossel bamboo, which undergoes dramatic shifts in resource allocation during flowering. To explore the mechanism behind these phenotypes, we investigated PHEFT1's protein patterns. Using YEST2 hybrid screening, we found that PHEFT1 can interact with itself and also with several PHEGF14 family proteins, specifically isoforms A, B, F, G, and H, as well as with PHEFD, a transcription factor homologue. We confirmed these interactions in plant using bimolecular fluorescence complementation. The BIFAC assays showed that PHEFT1 interacts with PHEGF40 in both the nucleus and cytoplasm, while its interaction with PHEFD occurs primarily in the nucleus. Let me now summarize the key takeaways from our study. PHEFT1 is a nucleus-enriched protein with a conserved structure of a functional FD homologue. It shows tissue-specific and photoperiod sensitive expression, with high expression in bulbs, stems, and late-stage inflorescences. Functionally, it promotes early flowering and reduces plant height and stem thickness in Arabidopsis, indicating a role in both reproductive timing and vegetative growth. In terms of mechanism, it interacts with PHEGF14 and PHEFD proteins, suggesting it acts through a conserved flowering gene activation complex to regulate flowering and morphogenesis. This concludes my presentation. Thanks for your attention.
Thank you, Vicky, and is there any also promote the flowering.
Oh, okay. Thanks for your question. Expect FT. Other flowering promoting gene includes MEDS box genes, COG, and FD gene, and so on.
Thank you. And also, so you can understand me, right?
What?
Can you understand me? I have a small question.
Okay, you say.
Most I miss the background. And I don't know what's the hamburgers if you have G1 in rice.
Oh,
Also, we should have HD3A.
HD3A is in rice and FT is in Arabidopsis.
Oh, yeah, the hamburgers.
Sorry.
They are both the hamburger, your G1.
No. Respectively, 就是 FT is only in Arabidopsis, and HD3 is in rice.
他们都是你的同学，这个就是你的同学。
对，就是一个单独在闽南界上，一个单独在水稻上。
In most of the ground, actually one. So which one is yours?
What?
但毛泽东有很多 FT。
对 ，yes。
所以这就是跟他同学到最近的 closest.
Yeah. This one?
I think the first three. 7.
Oh.
So you only yourself mentioned with the starving.
Yeah.
But why do you think the expression in the bar?
By experiments, we found it also can, 那个 yes.
Okay, sure. Because I missed the background. I mean, the postage.
Oh.
So there's no phenotype of this. Phenotype of the large element, you know, explaining hypothesis is anion frequency. 就是在你的转基础中，这个2 balance 是能够很差异的。你比如说，它有可能也有这个影响材料差异的这个功能吗？就是你现在观察到的好像只有开发方面，但是在两方面还没有展示。
对，因为我们就是主要还是侧重于它开发这一个，因为这是它最显著的一个功能嘛。
因为这个基因是挺好的 ，and I think there are many classification about that. I don't know whether you have read this. 应该看过50%，在毛谷的 FT 应该做了很多已经发表的东西了，所以为什么还要继续读？
This is my 那个 master sister do it.
You need to find some novel research 就是找新的观点，要不然它就挺好使。
Okay.
Thank you. Okay, that's the next speaker, Harris. Okay, so this topic is about managing the village, the garden. Oh, I don't know how to read it, process by near process, mostly viruses, right? It's not a virus.
Here I'm presenting my work, which is related to virus. Analyzing the cleavage of cardamom proteins by NA protease of cardamom mosaic virus. Cardamom, it's in Chinese Hou Dou, I think. It's the third most expensive spices, next to the it's the third most popular spices, and it's also called the queen of spice. It's mainly affected by the cardamom mosaic virus, which belongs to the Macrolora genus of Potibiridae family. The Potiviridae, it's the second largest plant infecting virus, which is similar to the Picornaviridae, which is an animal infecting virus. The genome organization and the polyprotein processing is both similar for the Picornaviridae and Potiviridae. So the Picornaviridae encodes the 3 proteases, leader protein, 2A protease, and 3C protease. These viral protease not only involved in cleavaging of the viral protein, it's also involved in cleavaging of host proteins in order to protect itself from the defense of the host machinery. So in coming to the Potiviridae, mainly the polyprotein processing is done by 3 proteases, P1 proteases, H3 proteases, and NIA proteases. The P1 proteases is absent in Maclovirus genus. So in Maclovirus genus, the polyprotein processing is done by HC protease and NIA protease. This HC protease has a cis cleavage activity, which means it will cleave only itself. But the NIA protease has trans protease activities, which means it will not only cleaving the itself, it also cleave the other protein for the transfection. So coming to the NIA protease, compared to the proteases of a picornavirus, there is a single report is available that the plant infecting virus not only cleaving the host itself, but it also cleaving the host protein to protect itself from the host diffusionary mechanism. So from the transcriptome data of cardamom, we have identified the cleavage sites of NIA protease in 8 different host proteins of cardamom. In this, I have worked in a prefoldin subunit 2. So the prefoldin, it's a molecular chaperone. In eukaryotic prefoldin, it's there are 2 groups, Archaea as well as Eukarya. For Eukarya, it belongs to group 2, and it has 6 subunits. Prefoldin 3 and 5 comes under alpha type, and 1, 2, and 4, 6 comes under beta type. This forms an heterohexameric complex to deliver the newly synthesized unfold protein to the cytosolic chaperone containing protein for folding. So the structure of this assembly in yeast is mainly it will form the gem-like complex with the beta barrel backbone with 6 coiled tentacles as an extended. There are also another group called prefoldin-like complex, which is similar to the prefoldin. The only difference is the subunit difference. In case of prefoldin-like complex, the subunit 2 in alpha type, subunit 3 and 5 is replaced by other 2 subunits like UXT and URI, and in beta type, the 4 and 5 is replaced by the PDRG1 and ASDURE. So among this, we mainly focus on subunit 2 because we have found that the cleavage site is present only in the subunit 2. So the main function of this prefoldin complex, if there is any disruption of any one of the subunit, the whole mechanism will be disturbed. So in C. elegans, they have found that the silencing of 1, 2, 3, and 6 cause the reduced microtubule and embryonic lethality and silencing of prefoldin. Similarly, the mutation in 5 and 1 leads to neurodegenerative effects and loss of nerve bundles in mice. And in Arabidopsis, particularly FDC6 mutation caused the impairment of microtubule and reduced plant size. And the deletion of prefoldin 2, subunit 2 and 5 cause the mitochondrial morphology and lethality due under the heat shock conditions. So my objective is to confirm the cleavage of prefoldin subunit 2 by the cardamom mosaic virus NIA protease and determine the cleavage sites of the cardamom proteins. So the methodology includes first amplifying the prefoldin subunit 2 from the cardamom leaves and cloning it into the vector and subcloning it to the protein expression vector, and to perform the performing in vitro and in vivo assay to prove the cleavage of the host proteins. In case of for in vitro assay, expressing expressing the NIA protease and BL2 separately and purifying the protein and perform the in vitro transprotease assay and performing the N-terminal sequencing. Similarly, for in vivo, we used to transform both NIA protease as well as the host protein, prefolded subunit 2 together in a single cell, a single BL21 and express, and we found the cleavage of the protein. So the results includes like RNA isolation was performed from the prefolding, and then initially we cloned the prefolding subunit without the site, and then further we cloned it with the BGL2 and ZO1 site in PGM-TEC vector, and further we subcloned it into the PACYC28 vector, which is a protein expression vector, and confirm the clone and express the prefolding further in our lab we already have the NAA construct, and further for in vivo transprotease assay we included we transformed both NIA protease as well as the prefolding subunit 2 into single BL21, and we found the expression of NIA protease and the cleavage of prefolding subunit 2, and further we have time of expression, and we have performed the different time interval like 25 degree Celsius for overnight as well as 37 for a degree at 3 hours, and we found the prefolding expression. And similarly for temperature standardization we performed the same experiment, and we found the expression was good in both 37 and 25 degree. And further after standardizing the temperature to perform the to standardize the solubilization of buffer, we have tried with different buffer like sodium phosphate buffer and urea buffer, and yes, and after confirming this I just moved to next. I have tried to amplify the another gene, Werke gene, which also has the cleavage sites. So initially we tried but not able to amplify, and further the Alpinia, it's an another plant species, belongs to same family. So we checked whether we predicted the Alpinia Werke gene, and further done the multiple sequence alignment with the monocot species, and we found the cleavage is present only in the cardamom and ginger family has the NA protease cleavage sites, and further I have amplified the N-terminal part and cloned it into the vector, and further it need to be work on Werke gene. Thank you.
Oh, I have a question.
Yes.
This dishes.
Cardamom.
Cardamom.
Yes.
Bamboo species?
No, it's not a bamboo. No, it's not bamboo. No, it's not. I have not done this work here. It's my master thesis work.
Oh, so it's still before.
Yes. Yes.
Okay, thank you. Let's go from the last one. Xu Jingxuan. So the topic is about the density dependent hybrid transpiration junctions and that's draws attention to most understands.
teachers, and classmates. As an undergraduate student, I am very pleased to share some research results with you today. This report is based on the research on the drought response mechanism of moso bamboo. We will expand from 3 parts. The frequent occurrence of extreme weather events is profoundly affecting the global forest ecosystem, and moso bamboo as an important economic and ecological tree species in southern China. Its drought sensitivity urgently needs attention. Global warming has increased the frequency and intensity of extreme drought events, and the drought has become one of the dominant factors leading to global forest decline and tree death. Therefore, exploring the drought response mechanism of moso bamboo has important practical significance. Why is moso bamboo particularly susceptible to drought? This is closely related to its structural characteristics and water physiological properties. Structurally, the stems of moso bamboo are hollow and thin-walled, without secondary growth, resulting in limited support and water storage capacity. At the same time, its roots are shallow, making it difficult to absorb water from deep soil. Physiologically, moso bamboo has high sap flow density and strong water and transpiration, leading to a faster water consumption rate. Under drought conditions, it is very easy to have an imbalance between water supply and demand, which is a core reason for its drought sensitivity. At present, relevant research at home and abroad mainly focus on the theory of stand transpiration and water resource balance. The core idea is to maintain the balance between water supply and demand under drought conditions by regulating the total transpiration of the stand, which provides a theoretical basis of our research. Regarding the drought response strategies of moso bamboo forest, existing studies have shown that moso bamboo forests are difficult to withstand short-term high-intensity summer droughts, and artificial regulation is required under severe drought. Under moderate drought stress, moso bamboo will adopt a variety of drought-resistant strategies, such as the conservative water use characteristics of old bamboos, water redistribution by roots and rhizomes, leaf isoprene emission, and active drought-resistant mechanism of roots. These studies provide references for our subsequent exploration of the role of density regulation. A larger number of studies have confirmed that density regulation is a key means to improve the drought resistance of forest. Reducing stand density can significantly increase forest volume growth and drought resilience, reducing drought-induced mortality by 2/3 in arid areas. Trees are more sensitive to competition and the impact of density is more prominent. Compared with high-density stands, low-density stands show better growth status and stress resistance under global warming and drought conditions. The chart on the right also intuitively shows the differences in forest drought resistance of remaining trees under different management strategies. In addition, density regulation can also affect the biomass of moso bamboo. The total stand biomass is an important indicator of productivity and is closely related to plant density. Sparse planting can change the individual biomass, while dense planting will reduce the average individual biomass. Therefore, the stand density of plantations can be reasonably determined according to market demand, which also provides a basis for our experimental design of setting different density gradients. Based on the above research background, the present study established 6 sample plots with different densities, each covering an area of 100 square meters. The monitored indicators are divided into 2 categories: physiological and environmental indicators. These indicators are used to comprehensively explore the relationship between density, competition, environment, and the
physiological characteristics of moso bamboo. The core framework of the experimental results first focus on planted utilized water resources. According to our hypothesis, the water use efficiency of the low-density group should be statistically higher than that of the high-density group, and the subsequent research will be centered on verifying this hypothesis as a core theme. We classify the 3 plots with higher densities as high-density plots, and the other 3 with lower densities as low-density plots. To qualify the competition relationship between plants, we use the Hegel competition index based on the Voronoi diagram for calculation. The calculation results showed that the average competition index of high-density plots was higher than that of low-density plots, indicating that stand density has an impact on the competition intensity of moso bamboo. Further analysis of the relationship between competition index and DBH shows that in both high-density and low-density plots, the competition index has a negative correlation with DBH. This means that the greater the competition intensity, the smaller the DBH of moso bamboo, and the correlation is more significant in the high-density group. Panels A, B, and C correspond to the 1st, 5th, and 10th days post-rainfall, respectively, demonstrating the dynamic association between sap flux density and the competition index in high and low-density stands. The result shows that the negative correlation between sap flux density and the competition index in high and low-density stands varies dynamically. High-density stands respond significantly in the early stage, strengthen in the mid-stage, and weaken in the late stage. Low-density stands strengthen in the mid-stage and remain stable in the late stage. It reflects the temporal differential impact of the combined competition jolt effect on water transport across stand densities. The spatial interpolation in results of soil water content shows that the higher the density, the lower the soil water content. In the plot, the soil water content in the areas where moso bamboo is clustered is usually lower, while that in the sparsely distributed areas higher. The figure on the right further shows that the soil water content of low-density plots is always higher than that of high-density plots, and the decline rate is slower. Leaf water potential is a key indicator reflecting plant water stress. The result shows that the leaf water potential of low-density plot is higher than that of high-density plots. The decline range of midday is larger than that of down, and the variation of midday leaf water potential among individuals in high-density plot is greater, indicating that the high-density group is more severely affected by water stress and has more obvious individual differences. Further analysis of the correlation between leaf water potential and the soil water content shows that down leaf water potential has a positive correlation with soil water content, and both density plots show that the higher the soil water content, the higher the leaf water potential. While the linear correlation between midday leaf water potential and soil water content is weak and not significant, it's worth noting that under the same soil water content, the leaf water potential of low-density plots is higher, and this difference is more obvious at midday. The determination results of stem hydraulic conductivity show that the 1st, 5th, and 10th days after rainfall, the hydraulic conductivity of high-density plots is higher than that of low-density plots, and there is no obvious difference between different days in the same density group. There is no significant linear correlation between the hydraulic conductivity of the 2 density plots and the soil water content, indicating that it may be more affected by the plant's own structure rather than the direct impact of soil water. The results of leaf relative water content show that on the same day after rainfall, the leaf relative water content of low-density plots is higher than that of high-density plots. Under the same density, it changes with the increase of drought days, and the leaf relative water content has a positive correlation with soil water content, indicating that soil water is an important factor affecting it. The results of sap flow density are consistent with the trend of leaf relative water content. On the same day after rainfall, the sap flow density of low-density plots is higher than that of high-density plots. Under the same density, it changes with the increase of drought days, and the sap flow density has a positive correlation with soil water content. The higher the soil water content, the greater the sap flow density, reflecting the higher activity of plant water absorption and transportation. To comprehensively sort out the connections between various indicators, we conducted a correlation analysis. In high-density plots, water indicators all have a strong positive correlation with soil water content, while conductivity has a weak or negative correlation with most indicators. In low-density plots, the positive correlation between water indicators and soil water content weakens, and weak negative correlations appear between some indicators. The correlation pattern of conductivity varies with density. Based on the preliminary results of this study, we will carry out two aspects of work in the future. First, based on the competition index data of individual mossel bamboo plants in the plot, construct a competition index heat map and conduct a comparative analysis with the soil water content interpolation map and a Voronoi diagram to further reveal the relationship between spatial distribution, competition intensity, and soil water. Second, based on the analysis framework below, construct a structure equation model to deeply explore the path mechanisms through which stand spatial structure and soil water jointly affect the water physiological traits of mossel bamboo under high temperature and drought conditions, so as to provide more accurate theoretical support for the rational management and the drought resistance regulation of mossel bamboo forest. This is the end of my report. I appreciate your valuable feedback and guidance.
Thank you for the presentation. And is there a question? Well, I also ask for a later question is, what's the state and you choose, and you will take your sample, you also can go. Yes.
Why in the summer?
Because the different season has a different soil
water content. We do this research in Anji in August.
August, that means we are in the autumn. August, in the autumn.
Summer.
Autumn season. Okay. Anyway. We do have the distribution of which example on the map. How far that means? 30. Yeah. Yes, you just example as the energy map.
Yeah.
And what's the different place are you covering? Compare different mossel bamboo in a different place, just not in the energy.
No, we just make our research in Anji.
Do you have some distribution? Take some sample as energy, as in the map as a map distribution. I think you can add a map. That you take some samples in the energy, the distribution of the mossel bamboo. I think actually under when I translated to that sense，意思就是说，如果你要是发表相关的文献的话，我觉得比如说你做的是空间密度性的分析嘛，用的是 WRAM 的那个，就是 GIS 当中的一个密度分析的一个图，我觉得需要在前面去补充一张那种，比如说安吉的一些有一些样本的一个图纸，就是比如说你去现场。采集的一些样本图纸，
是类似于，
因为你做的是密度分析。
哦，我在下面这个有那个取样，就是给它打了点。
就是说需要有一张总的一个密度分析图，比如说你的一个类似于前面的一个分布图纸。因为我看你也是用GIS做了一些相关的分析，就是如果你要是去发表文献的时候，我建议你前面有一个总体的一个这样的一个图会比较清楚一些。
Thank you.
Okay. Thank you for this presentation. So you have a question? Yes. Regarding the comparison of water with physiological characteristics between high density and low density instance. We can know the results without conducting experiments. So what is the significance of your research?
Thank you for your question. The water physiological characteristics is just one part of our research. And our purpose, the final purpose of our research is to look into the competition and the soil moisture interact to affect the water physiological characteristics of mossel bamboo. And thank you for your advice. In the future study, we will emphasize the parts related to competition and environmental factors. Thank
you. Thank you. So, it's rather the last one. Certainly we all has managed for a long time and it's already finished. Thank you.
陆老师，你是在这里吃过饭，还是去那个当时酒吧，就是那个酒吧？
我都可以。
你看你如果那边要去的话，你跟我们
我就在那边走，都可以。那就啊，行，我就跟我团队那里。我不是这样的。反正下午2点要开。
这个可以的，这个可以。哎，这个数。
谢谢。
这个是谁的 U 盘呀？
有点可好，刚才我
是主任呢？这个可以关了呀。
我把这个东西完，拜。 磁吸的都是我们的，磁吸的有14个是你们的哦。
那个实施的，你们要拿，你把你们那些拿回去就好了，别到时候我就把它带走了。
对哦，所以说我先帮你收下，你剩下我不要了。
你想把我们这里借的就回去，你给他
现在
来。你晚上吗？我们好，这边我给你来一个，这个5万是谁呀？我告诉你，就是可以啊，我会组织别人还有哪？啊，不知道那个，那个，姐，你吃过了没有？
我吃过了。
啊，你。
那我先去报个一下。
第二个，你看这个，我们现在就开始1:45左右你就到了，或1:45。好，那可以这样，这个就好。等我稍微取个1:40，到这里。现在已经1点，再
谁？大家，不要紧张，大家。我在讲不是其他的，不行。更往前。一个对吧？一个是3，一个是2条。
中间几行，中间用英文太长，其实它这个文章不长，而且挺好看。他最后边这个提出了一点错，就是说，要是不能回答那个你自己图对的，他们这样不行。
对啊，我是那个。
附加题是。
5啊，这就是输进去。这6就是输一下，就是输进去。修一修吧。
最起码一天2个2个啊。一天打完不是人全来。
这个不能关是吧？嗯。
PPT 那就不搞了。或者这方面，这方面讲会。
对，那是2天里面取一个最高的，作为那个一等奖。
因为我以为。
我在其他地方是能去啊。那个没有甚至说。
哦，你看这个哦，这吃在里面，它写出来。那船上要。
啊，我你在说什么？啊，我发给朱雨晓了。我跟邹老师说了，我发给朱老师了。
这个都是人工八八，是不是？大学。
我做了，我发给王志康，然后不知道为什么。哎，志勇，这个电脑的垃圾箱在哪儿？
回收站吧，没有回收站吗？
哦，回收站，我的Windows 电脑好久没用了。
啊，回收站，这个，
哦，他们这个主旨报告都要先删掉。
哦，
哇，这么多回收站。
对啊，今天啊很多
它一清空就是一下全清空。
啊，不要要清空嘛。
回收站啊，这是。
对啊，
有哎，对他们都知道的，都是在颁奖典礼里宣布呀，肯定会参加。如果觉得自己会得奖，肯定会参加。不用通知，不用通知，不用通知。呃呃，
这个能删吗？
这个能单独删除？
可以可以。
就这样删了。
嗯。
然后 developing 这个也是。先把这个投出来吧。
是不是下午那个？不是，是不是下午那个说？要做的。啊，对对对，后来这个我这个有情况不到达。对，24。现在这个香港东西方案的活动啊，活动空间方案，对，他们对这个会在，你看这个活动空间方案有。
你拿到两个都，有区域的，区域的，这样的话，因为空气的，不感觉到。这是的，可以说是但是不要很好，就是对方我当然有听啊，我知道了，说话了，就是那就吃吧，我让回来吃的话，你吃了，快吃点别的饭。还有，都不要。我发挥这种贴心方法。哎，好，这种品质算了，他对那个地方小区的问题。
我们一能涨几角？跟这二三来讲，它差的分太多了。他们快满分了。
可以让没力强。
对，当时正好今天晚上要生产，就想让新闻觉得，那讲一定是最重要。但是它就很让人猜测它到底是不是。但其实我感觉跟他做这个主持有起来，我觉得他很直啊，就是。我这个说的这句，我都听完了。跟他来个致谢，一会儿去台上就给他来。
真的，你不知道那个人吗？你的机箱。
that's what you know. 不知道，一般各厂家没有，一共没有，没有自己的。这个好，小那个。 都得去，不清楚都得去。刚讲的人，对，好的，潘博，你今天做报告了是吧？你做报告了是不是？对。好，那你等着。我也遇到。之前我在旁边。我叫他前人。你在旁边。我跟你非常玩，我和安达我也一样是吧？我们吃饭，对。那我给我点慢一点，知道了。啊？不是，这个结果是在发布群里的，结果。那我们现在，那我们不说了，我们先不做。对。最终你要确定好的要如何？那那个会场的人练，他们是给我看还是那个图片我看？那自己搞一下吧，是吧？
一年是发给我们看到这个对，是发给咨询发给我们
现在王一在群里面吗？在那个微信群里面吗？
他不在，我给他电话。
本来是很简单的事情，我不是因为这交通那里说，那我不干了，我回去睡觉了。那我你像我给的这个信息，不知道他，你看我这东西来全都是，除了一个是校外的，其他全都是摄录影，那很多，而且校外的那些你说这些都是录影，对。对，
现在就是往这边我们是科研
那几个是消防同事，那我给准备正好一个。
对，对，因为那个一开始跟对吗？那个全部评判，那估计就给我那个不弄
我来，那边是我来。
但也不知道在你给厂也在哪里，对吗？
我来，这个
这个都是跟任务
第一个是什么？
关键是你看少了，也不能一看的，
全部就想按那个，一代老师。
不是INBAR。
开始跳队，二十分大家都没有办法。
老师在给你讲座，真的他的那个思路是这样，人家要做那个主持人嘛，你不是嘛？你到一到他呀，上来一边讲完吧，对，在里面，不知道，在班上说，班级不爱讲。对呀，得看领导什么意思了。但你是
选他什么多了？
哎，但是差别贵，差别贵，一点都不。全换线上，我这有全。第一是在线上，但10月1半大概12月半，但它不是来自他们的。然后那个前面溜溜，因为第二天是在线上，线上那个哦，你我这个信息，这到底想到你不要说出来嘛？那你给你的办法，之前我们说了，你们产业这个还有跟着抢，咱们有一些是跟你这个老公，咱我现在我上六级，
我刚才看见他，对，
吃饭费，
吃饭费，对，先给个吃饭费。拍点，他现在。
他们说的有什么用啊？
哎，我讲，对，都哎，我们都快等到我去那边，我们就才好，就回来了。你说你这没多强啊？家庭里太大，也是一个。 它那个是因为显示它管理的很好，换一个人，它那个团队是英语太少。
哦，真的。那是谁？我看你挺会的，挺好。我几期后他回来晒了，也
好像怎么去过？
一开头是什么？好，左边是。电脑有点慢。这个对了，对了，然后我共享。这些有东西应该还在电视机里。哎，我是把放大。然后翻译的话，还是用这个翻译好。你看，我们一个搬到这里就好了，然后这里面就是那个颁奖了，颁奖的后面的还没做好，到时候我再考一个回来。这里比较不打就好。
ወይ ሰላም ነው? እግዚአብሔር ይመስገን። እንዴት አደርክ? ደህና ነህ? አለ ይመስገን። ጆይን አድርጋል። ዙሙን ጀምሯል። አሁን ዛሬ ነው የጀመሩት። ትናንት የለም እምቢ ብሎኝ ያው ዛሬ ልከውልኝ አሁን ገባ። ግባ እየተጀመረ ነው። መሰለኝ ይሄው ዲሴምበር 3 እስከ 4 ይልና 2025 አሁን ጀምሯል ቻይና
I'll start. I can start. I'll start. Okay, I will start. Okay, hello everybody. Welcome to our closing ceremony of the 3rd International Young Scientists Forum for Climate Change, Sustainable Development through Bamboo Resources. And first of all, we will have a keynote speech for you all. We have our distinguished Professor John Ennis, who's a professor at Faculty of Forestry at University of British Columbia. And his keynote speech will be the sustainable development of bamboo forests. Okay, welcome.
Okay.
This works.
Sorry?
This works.
Okay, good. So good afternoon, ladies and gentlemen. It's a pleasure to be here. I've been learning a great deal about bamboo over the last day and a half. We've heard some really excellent presentations, both from those of you present and from people who have joined us from around the world. So this has really been a spectacular event. Probably more papers presented about bamboo than you'll find anywhere else in any other conference. This afternoon, I've been asked to give a sort of a wrap-up talk and maybe just remind you about the importance of bamboo and why we're here, why we're considering it as being such an important material. I'll start off by outlining a little bit about UNCED and the Montreal process. We'll talk about criteria for managing bamboo forests sustainably and just run through some of the new products and the new markets that are emerging for bamboo products. A lot of the talk about sustainability can be traced back. In some cases, actually, we can trace sustainability talks to over 1,000 years ago or even further back in time. But really what I think was the catalyzing event was the 1992 United Nations Conference on Environment and Development. It's also known as the Earth Summit. And it was important because it was the first of these big things, big conferences. They're now really common. I mean, there are 50,000 people were in Manaus at the COP meeting. Some of the other meetings have had 80,000 people present. But in 1992, that wasn't happening. And so getting 100 world leaders together, as well as a bunch of others, was really quite an achievement. The themes that they talked about then were actually themes that we're still talking about today: sustainable development and global environmental change. The concerns also haven't changed all that much. In the northern countries, people were concerned about the loss of biodiversity, about climate change, about the hole in the ozone layer over the Antarctic and then in the Arctic, and associated UVB and skin cancer, and rising sea levels. We're still talking about rising sea levels. We're still talking about climate change. We're still talking about the loss of biodiversity. But we did manage to do something about the hole in the ozone layer. So those international collaborations do actually eventually sometimes come to fruition. In the southern countries, we were talking about poverty alleviation, about food security, about economic growth. And guess what? We're still talking about poverty alleviation, food security, and economic growth. What UNCED actually achieved were a number of important agreements. There was the Convention on Biological Diversity, with its aim to conserve the biological diversity of the world. There was the Framework Convention on Climate Change, which was to establish mechanisms to combat global change. There was the Convention to Combat Desertification. Now, unfortunately, despite those conventions, we're still losing biodiversity. We still face climate change, and we're still trying to combat desertification. There were also other actions. There was the Agenda 21, which was an action plan for sustainable development that probably many of you have never even heard of now. It seems to be largely forgotten. In that document, 351 pages, bamboo was mentioned once. Only once. Forests were mentioned 289 times. Water was mentioned 618 times. Bamboo once. I think if we were to write Agenda 31 today, bamboo would appear much more prominently. At least I hope it would. We did have a statement of principles on forests in 1992, which was a non-legally binding statement of principles about sustainable forest management. Again, no mention of bamboo in there. There was the Rio Declaration, which was a set of non-binding principles to guide the course of human development. And again, bamboo not mentioned. In other conventions, we've had the CITES. No bamboo species were considered to be endangered, partly because conservation assessments had not been done of bamboo. There was the transboundary air pollution, not really relevant to bamboo, and the World Heritage Convention. Again, I guess there might actually now be some World Heritage sites where bamboo is involved. The early 1990s saw a series of guidelines for the sustainable management of forests, but they didn't mention bamboo. So we had the guidelines for sustainable management of natural tropical forests, criteria for the measurement of sustainable forest management, guidelines for the establishment and sustainable management of planted tropical forests. Nothing on bamboo. The ITTO played a big part in many of those. They have been producing forestry guidelines. Those guidelines have not necessarily helped the sustainable management of tropical forests. We're still facing substantial deforestation. The northern countries, after the Rio, and in response to criticisms that they were deforesting, produced the Conference on Security and Cooperation, which had a meeting in Montreal to develop criteria and indicators of sustainable forest management. It dealt with temperate and boreal forests. I was at that meeting. I know that bamboo was not mentioned, not once. That evolved into what's known as the Montreal Process, involving the world's temperate and boreal forests, including Canada, USA, and China, does not mention bamboo in any of those criteria. We saw a whole series of other processes associated with the development of criteria and indicators of sustainable management of forests. And you can see there a bunch of them: Helsinki process, the Tarapoto, Le Patrick, the Dry Forests Asia, ITTO, Dry Zone Africa, Near East. No mention of bamboo. Something's going on. Do you see that pattern? When we look at the definition of sustainable forest management, it talks about the stewardship and use of forests and forest lands in a way and at a rate that maintains their biological diversity, productivity, and so on. Doesn't specifically mention bamboo, but bamboo can be included in that. Bamboo is an important component of forest ecosystems, and therefore it does fall within this broad definition produced by the United Nations Food and Agricultural Organization. So where are we with bamboo? We know, as all of you do, I'm sure, subfamily of the grass family, the Poaceae, round about 670 species in round about 125 genera. Please don't say an exact number because it's always changing from day to day almost. Even the genera are changing. There's only a small proportion that are economically important. 44 species are listed by Long et al., and there's been a further 16 species that have been added in an additional list. So only a small proportion of those 16, approximately 1670, are considered to be of importance economically. In terms of where those lie, bambusa, dendrocalamus are important. What you can't see is Philostachys. It's just off the screen for some reason. I'm not sure why. Also, obviously important, Philostachys edulis or mozo bamboo coming out there. Also lay bamboo. Just to give you an idea of what we're talking about. These are the examples of some of our economically important species. We believe that one of the reasons that bamboo is not included in some of those international figures is that it is being underreported. About a month ago, the Food and Agricultural Organization released the Global Forest Resource Assessment 2025. In that, 158 countries ticked the box that said, do you have bamboo? But only 33 reported on their bamboo. We know that in those 33 countries, there's about 30 million hectares. India and China account for the most, 11.8 million hectares in India and 8.5 million hectares in China. These are the latest figures published only a month ago. In terms of its distribution, again, we know where it occurs. It's planted beyond its natural range, but it does occur in a much wider area than some of the maps I've seen presented in the last day and a half. In terms of its global area, bamboo has increased by 8 million hectares between 1990 and 2025. However, since 2020, there has been a decline in the reported area of bamboo. Now, that may be a real decline, or it may simply be some countries that have failed to report. So not every country, as I mentioned, is reporting. There have also been significant, but in many cases undocumented declines in the quality of many bamboo plantations, largely due to a lack of management. And that's something that we are paying more and more attention to. It's not just the total area, but it is what are those areas actually like? Is the bamboo vibrant and growing well and managed well, or is it in decline? This is an example of the sort of areas. And those of you going to Anji tomorrow will actually go past that area of forest. So what principles should be adopted for the sustainable management of forests? Because it's a non-timber forest product, it is actually covered by most of those sustainable forest management schemes I mentioned. They're not specifically mentioned, but it is in there. If you look carefully, you'll see references to non-timber forest products, and bamboo can therefore come in under that. Those schemes actually show many similarities, and the Montreal process is the one I'm going to go through and actually say, how well does this fit for bamboo? But you could take any of the others. They're very similar. The Helsinki process, almost identical to the Montreal process. So criteria 1, conservation of biological diversity. Does bamboo contribute to that? The answer is obviously yes. There are a whole lot of species that are dependent on bamboo. Obviously, the iconic giant panda is one, but the red panda, another one. There are many important species that consume bamboo. The mountain gorilla, the Indian elephant, the South American spectacled bear. And there are actually several species of bamboo lemur in Madagascar that are entirely dependent on Madagascar. It forms an important habitat that many organisms use either 100% of the time or at least some of the time. And we know that, for example, 5% of bird species in the Amazon live amongst bamboo. There are many species that are entirely restricted to bamboo forests. And they include some pretty strange animals like the mountain bongo of the Central and East African mountains. How many of you have heard of a bongo? I don't see anyone putting their hand up. That's what a bongo looks like. The reason you probably haven't heard of it is nocturnal, which is why the picture is taken at night. You only find them around at night. But it eats bamboo. It lives in the montane bamboo forests of East Africa. There's an example of one of the bamboo lemurs in Madagascar. There's a bat that only lives in bamboo forest, the lesser bamboo bat. There's the large bamboo rat, depends on bamboo forests, occurs here in China and also in other parts of Southeast Asia. There are birds like the spotted bamboo wren in the Atlantic forest, that's the coastal forest of Brazil. Or the Chinese bamboo partridge. Have any of you seen that bird? Occurs here in China. Any of you seen it? You're not spending enough time out in bamboo forests. Or the black-throated parrotbill in, I think this is actually in Nepal, judging from its name. There are even bamboo leaf frogs, again, a Chinese species, and bamboo snakes. So a whole range of different things. A fungus found in Northeast India, bioluminescent. It occurs on bamboo. So we're finding more and more things. There's lots of species still to be found that are restricted to bamboo. Criterion 2 of the Montreal process is about the maintenance of productive capacity of forest ecosystems. Well, we should be concerned about the maintenance of productive capacity of bamboo ecosystems, whether they're natural or, as in this case, they're planted bamboo. There's been a lot less work done on bamboo productivity than most commercial tree species. We do know that bamboo tends to be more nutritionally demanding than forest trees. So we need to manage them with fertilization if we're wanting to maintain productivity. They've also got high water requirements, and in some cases, irrigation may be necessary. Other things, that management aspects that are required to maintain productivity include regular weeding. So we've got a lot of things to do, and unless there's profit to be made, it's very difficult to convince anyone to manage the forests, as I think we're finding here in China. There are some existing guides to bamboo management, but a lot of them are fairly old and they require substantial revision. And in particular, we need to actually look very carefully at the guidelines for managing clumping bamboo. A lot of clumps have been allowed to become congested and they are losing their productivity. We need pruning for some of the thorny thickets. I've got Bambusa bloomiana. I thought I corrected this morning, but obviously the wrong version got loaded up because that's now Bambusa spinosa. I know I had to say that because I knew someone would correct me in the front row here. Bamboo shoot reproduction requires more intensive management than culm management, very labor intensive. And having seen what goes into the shoot production around here, with all the extra work that's required, particularly with the rice husks that are spread on it during winter to try and encourage early growth, that Renyi Gui has shown me. I was astonished at just how much work must go into these plantations. So can we maintain productivity? It's going to be a challenge. How did I do that? Am I going forwards or backwards? I think I'm going forwards. So there has been quite a lot of work done on some species, but we're still very short on work done on all the other economically important species. We don't really understand the genetic variation within the natural ranges of many species. There's been a lot of work done in Ethiopia on highland and lowland bamboo and the different land races that you find there, but we don't see that sort of work going on elsewhere. We also know that there are changes occurring in some of these ecosystems. This is an area that, again, Renyi showed me last year in Sichuan, the Shunan Bamboo Sea. And you can see here, there's something happening here. It's not pure bamboo. We have broadleaf species coming up through the bamboo, probably related to lack of management. In terms of criterion 3, maintenance of forest ecosystem health and vitality, obviously bamboo, we should be doing that too. We should be looking after the health of bamboo ecosystems and the vitality of bamboo plants. A big concern is the die-off that occurs with woody bamboos after flowering. We see annual flowering, we see sporadic or irregular flowering, and the one that really worries a lot of farmers is gregarious flowering, when all the bamboos in an area will die in a particular year or over a period of 2 to 3 years. So as an example of one of the species that flowers gregariously, Thamnocalamnus spathiflorus. And bamboo mortality. I was in Karnataka 2 years ago, and all the bamboos over a very large area is looking like this. It's dead. And you go to farmers and say, you should be planting bamboo. They say, well, why? It dies. So we need to understand how that works and how we can avoid having all the bamboo dying at the same time. What sort of, does every species respond in the same way? Or can we find species that does not die in synchrony with all the others? We know that annual flowering is in the herbaceous bamboos. They tend not to be important economically. We know that some bamboos can actually both flower gregariously and sporadically. Guadua angustifolia is a good example of that. That gregarious flowering, which is so important in terms of the economics, the flowering intervals vary. They can go from 3 to 150 years, depending on which species we're talking about. Even within species, we find very variable timings, and we don't really understand, as far as I know, what is actually triggering the flowering events. There's various theories, but it's probably different things in different places, even with the same species. So Bambusa vulgaris, for example, anything between 80 and 150 years. Dendrocalamus asper, anything between 30 and 120 years. The Bambusa vulgaris, actually one of the longest flowering intervals, up to 150 years. But when it does flower, it's a disaster locally. In terms of bamboo health, like all organisms, including us, it's subject to damage and disease. There's been over 1,100 fungal species recorded on bamboo, and we suspect there are more than 9,000 fungi. There's a lot of phytopathogens like Stereostratum or Ceratosphaeria that affect bamboo. And as we move and try and develop plantation monocultures, we increase the risk of disease and economic loss. So we need to be really careful and understand this much better than we currently do. When we introduce bamboo from one area to another, we need to have a really good understanding of the species requirements and also of the likely pests and diseases that might affect it. There are some species that appear to be adaptable. Bambusa vulgaris, Bambusa balcua, or Dendrocalamus strictus are all good examples. But others have much narrower ecological requirements, especially for soils. But we don't really understand all of that. And there were a couple of papers in the last day and a half about ecological requirements. So I'm glad to see there's work going on. Dendrocalamus strictus, actually a species with very broad ecological requirements. And in fact, it can grow in semi-arid and arid environments, particularly when planted with other species. However, drought remains a big risk for it and for many other bamboos. And again, I was pleased to see a paper looking at drought and drought tolerance of bamboos. I think that was yesterday. Criterion 4 is about the conservation and maintenance of soil and water resources. Bamboo plays a big role here, and it plays probably an increasing role. I've got one of my students is actually looking into this in the role of bamboo in soil conservation in southern India. It's important for combating soil degradation. And again, we heard papers at this conference about that. The rhizomes work well to bind soil together, reducing the risk of erosion. But I have also heard that in some cases, it can increase the risk of landsliding. So we need to be really careful how we understand the links between the root systems and soil stability. Some of the figures that have been estimated are pretty impressive. So for example, the root systems have been estimated to extend 100 kilometers per hectare. That's pretty spectacular. In terms of restoration, there have been work by INBAR and others that have shown that we can actually restore areas. And again, we saw a paper about that in this conference. Bamboo can, in the INBAR study, result in 25% less water runoff and a reduction of soil erosion by 80%. So we're seeing huge benefits, and it's being documented and quantified, which is really important if we're going to persuade policymakers about the importance of using bamboo. Criterion 5 relates to the maintenance of forest contributions to global carbon cycles. And I think one of the things that has emerged in recent years is just how significant bamboo is in terms of carbon and carbon cycles. And a lot of that has been done by ZAFU, Professor Guomo Tsu, leading figure in this area. We talk a lot about the rapid growth rates of bamboo, and we equate this often with the high potential for carbon sequestration. The figures that have been produced from here suggest that 1 hectare of mozo bamboo can sequester between 4.9 and 5.5 tons of carbon annually. That's more than many tree species. However, if we're really going to understand how important bamboo is, we need to do more life cycle analyses of bamboo products. How long does bamboo scrimber actually last? Dai Changping gave a talk about bamboo scrimber and talking about how important it is, but how long does it actually survive? In terms of total ecosystem carbon in bamboo forests, can be anything between 94 and about 400 tons per hectare. How does that compare with timber? Timber tends to be about 100, maybe somewhere between 100 and 700 tons. So some timber forests contain more carbon than others. Plantations, on the other hand, tend to be less, which is where the comparisons have been made. The comparisons have been made between, for example, mozo bamboo forests and Chinese fir, Cunninghania lanceolata, or Pinus masoniana, mason pine forests. INBAR has actually calculated that if we have an additional 10 million hectares, we could be saving over 700 gigatons of carbon dioxide within 30 years. Just to put that in perspective, that would be equivalent to the savings made by 300 million new electric cars in the same period. Which one is government pushing? I think the Chinese government is probably pushing electric cars more than it's pushing bamboo plantations. So just to give you some figures, actually Philostachys edulis, mozo bamboo, is one of the lower estimates. We see higher estimates produced for Philostachys bambusoides, or Bambusa oldhamii, or Bambusa bambos. Up to 24 tons of carbon per hectare per year in the case of Bambusa bambos. Really, really significant. When we look at combined carbon sequestration and carbon emissions reduction through product displacement, when we're reforesting grassland with giant bamboo, either mozo or guadua, we do see big improvements, big changes. Those orange stripes at the top, total carbon sequestration, carbon emission reductions, up to 322 tons of carbon per hectare. We're also seeing bamboo being used for bioenergy, really important because it's replacing some forest fuels. It can also replace coal. So the reason it's important for forests is that one of the causes of deforestation is cutting for fuel. If we can replace that with bamboo, we actually have a double benefit there. We know globally about $60 million of bamboo charcoal and briquettes are exported annually. We don't know how big the domestic markets are because it's one of these informal products that isn't adequately reported. Right now, it's not widely used in commercial operations, but there is a lot of research going on about how we can actually avoid, for example, forming the clinker that damages furnaces when bamboo is burned. Bamboo Energy in Japan, they've actually worked out how to do that. They blend bamboo with Japanese cedar bark, and that raises the melting point of the ash, so they don't get the clinker buildup that damages the furnaces. As I mentioned, we could actually be using bamboo in many cases to replace wood for domestic fuel. And if we look at what has been calculated in sub-Saharan Africa, where bamboo wood fuel is still incredibly important, in some cases up to 90% of the energy generated in a country comes from wood fuel, we could about 9 million tons of bamboo charcoal could actually replace 64% of the wood that is cut for charcoal production. Just to give an example here, this is in Kenya. These are charcoal sacks cut from the acacia forests. The acacia forests are being basically destroyed because of the demand for charcoal. This charcoal's on the roadside. That charcoal will probably ultimately end up in Somalia rather than in Kenya. An example of the charcoal production plant here in Myanmar. Again, it's been using timber. You can see the timber stacked there, and the small cases. You don't see very many big trees in the background because they've all gone for charcoal production. Yet bamboo could be used, and we see it increasingly being advertised. This is just one taken the other day. The sixth criterion of sustainable forest management that we can use for bamboo also is the maintenance and enhancement of long-term multiple socioeconomic benefits to meet the needs of societies. Bamboo tends to be grown at small scale. It's often used in arguments about raising incomes of rural people. We heard a figure, 2.5 billion people depend on bamboo. That was cited during this meeting. I actually don't believe that figure. It's like how many people actually depend on forests. Someone invented a figure 10 years ago. They said it at a UN meeting, and it's been quoted ever since. The evidence is pretty limited. So I'd be very careful about some of those broad statements about how many people are dependent on either bamboo or forests. However, we do know that bamboo forms a really important part of local people's economies and livelihoods. We can link it to ecosystem services. We know that bamboo can be favorably compared with many other areas. So, example, what this shows is if we compare bamboo used for bioenergy than natural forests, it has a higher value than the wood does. It's a kind of a complicated diagram. Again, we can look at it for other aspects: groundwater recharge, water purification. These are all the ecosystem services. All the H's mean that bamboo is better. And again, the last one, for a few of the other ecosystem services. In China, China has demonstrated what is possible. Bamboo has been strongly supported in China by federal, provincial, and local governments. It's seen as an important instrument to support sustainable socio-economic development. We've had clear government involvement with the development plan for the bamboo sector in China, 2013 to 2000. We've had a document called Opinions on Accelerating the Innovation and Development of the Bamboo Sector, related in 2021. In terms of its value, again, we've got to be careful with some of these figures, but the sector has been valued at about $35 billion a year. It has been estimated to employ over 15 million people, 14 million of which are farmers, and the area has involved more than 7 million hectares. There's a strong belief in INBAR and elsewhere that other countries could follow this example. And those of you going to Anji tomorrow should really listen to what the Anji example can tell us. There's some really important lessons. There was a paper this morning given about that. There have been publications about it. Anji really is a wonderful case study, and it's just down the road from here. Bamboo contributes in all sorts of ways to this criterion. Bamboo tourism is growing in importance. This, I think, is in Anji. If you've got adequate supply chains that you can transport bamboo from the forest to where it's actually used, that really is important and helps. It's also a source of employment. There are truck drivers involved. We see bamboo being used in innovation in architecture. Some really nice examples here for exterior use of bamboo, but also the interior use of bamboo. Lots of new techniques being developed. There's a lot of people working on building design, a lot of people working on things like how to actually join bamboo together reliably. We eat bamboo. There's work going on how we scale up that process. So it's not just a matter of a farmer producing some shoots, selling them in the market. We're getting industrial scale development of bamboo shoots now. Producing products like these that are both consumed locally, but also exported. I can buy packaged bamboo shoots in Vancouver. In fact, a lot of our students seem to really like them. There are a whole lot of new markets are being developed for bamboo products, whether it be healthcare products, cleaning products, the packaging for cleaning products, lots of innovation occurring today. Some of the skincare products associated with bamboo. We're finding more and more things. And we have an expert in the audience here in these areas. The market for bamboo clothing is actually exploding at the moment. How many of you wear bamboo clothes? At least some of you should. I do. Shui does. That's it. Oh, yeah, we're seeing a few people putting their hands up, but no one from China. What's going on? It's all made in China. Sorry? No one would put their hand up. I have a bamboo t-shirt. I was wearing a bamboo t-shirt first thing this morning. We have other products that are coming more and more. So carbon is being used, bamboo carbon as a purifier for water. Incredibly important today as water problems increase around the world. And we can eat things associated with bamboo. I put this up for those of you who are not from China. How many of you have eaten these? How many of you want to eat these? Or even better, bamboo rats, which are now farmed on a fairly large basis. Sorry? I actually don't know where that photo was taken. I took it off the internet, I have to admit. This is much better though, bamboo beer. I think most of us would be happily eat that. Finally, criterion 7 is the legal, institutional, and economic framework for forest conservation and sustainable forest management. And this is really, how do we get bamboo into national policies? It is working in some places. This is the Republic of Kenya, has a national bamboo policy. We're seeing more and more countries beginning to publish bamboo strategies. As we heard I heard this morning in one paper, there's a lot of resistance in government to bamboo. A lot of civil servants still think of bamboo as being something substandard, or it's a weed, or it's grass. We don't need to be bothered about it. But more and more, we are seeing it. So the first was in China in 2013 with China's national plan for the bamboo industry. But we have other examples like the Uganda National Bamboo Strategy and Action Plan, Kenya's one that I showed you, Ecuador's National Bamboo Strategy Guidelines for Green and Inclusive Development. So more and more countries, but not enough yet. And they're not converting necessarily these policies into actions. There's some examples. So we have there the Kenya one I showed you. Tanzania has one. And I think that's Ecuador, the last one. Yeah, Ecuador. So we have these 7 criteria of sustainable forest management that we can be applying bamboo within. Difficulty is often that small-scale farmers never heard of the Montreal process. They don't care about it either. So how do we get those small-scale operations to actually start thinking bigger? And there are actually some nice examples, and guess where? Angie is again an example of this, where we have farmers coming together into cooperatives and ultimately having a single organization that is managing the forests. There's also a lot of other forms of cooperative management that we could be taking from forest examples and applying within the bamboo context. So in conclusion, bamboo is growing in importance as a global resource, even if the area of bamboo seems to have declined a little bit in the last 5 years. It produces numerous ecosystem services and provides a multitude of goods. Someone's mentioned 10,000 products. It's probably more than that. Its importance is widely underestimated, particularly by governments and by land management agencies. However, gradually seeing more recognition of the importance, and we now have this new journal, Advances in Bamboo Science, devoted to bamboo. It provides an opportunity for South-South and South-North collaboration, especially multilateral cooperation, such as is done by the World Bamboo Organization or INBAR. And so I'm actually really positive about the future. I think bamboo is a product that is going to grow and grow in importance. You're working in it. You're lucky. You're at the beginning of your career. Everyone's sitting in the back of the room. You've got great opportunities ahead of you if you manage those opportunities correctly. You just got to make sure that you do. Got to make sure that you actually can be an advocate for bamboo and create the opportunities that are out there if you just get to grips with them. And I really hope that you will do. What I've heard over the last day and a half are numerous really, really good studies, but not necessarily communicated very well. There's a lot of you need to practice your communication skills. And I'd be saying this to exactly the same words if I was standing in front of a group of Canadian students in Canada. It's not just language. It's your communication skills overall, the way you prepare a presentation, the way you give it, that's important. And I really hope that this conference will have helped a lot of you move forward in that direction. If you've given a presentation this year, doesn't mean to say you can't give one next year. I know there's a few of you here who presented last year and haven't presented this year. I don't know why not. You could have. And maybe you would have got one of the prizes that Chanyu is about to give to everyone. Okay, that brings it to a close. I think I'm actually still the moderator. So I believe our next stage is to give some awards out. Okay. So. Okay. Good.
Thank you so much, Professor John Ennis, for this comprehensive overview of how do we manage the bamboo forest and bamboo products sustainably. And I think as a Western scholar, you're more knowledgeable than 99% of the Chinese on bamboo. Okay, now I would like to invite distinguished Professor Nimala to announce the names of the awardees. She is the distinguished professor from the Department of Botany of the Punjab University.
Thank you. Good afternoon to all. And first of all, I would like to thank Professor Inns for his wonderful talk about the whole program also, and also the help and all is provided. Of course, ZAFU is there, but it has been wonderful. And you summarized the whole concept and you give some beautiful pictures, which for us, some were new to us, and we got some new information. We always learn. We may be professors or maybe scholars, but every day is a learning day for us. And it is with great pleasure that I'm going to announce the Best Poster Awards. And I would like to invite Dr. Merdelin to help me to do the honors. Dr. Merdelin from Philippines. Come. Yeah. Okay. So.
Give money?
Yeah. Why is it not moving? Okay. So yes, everybody has worked a lot. Everybody is presented the best, but they say they're all winners in that sense. And just a matter of some difference. So I call upon Noor Atira Anu Abdul Tab. I hope I'm pronouncing good. University of Malaysia, Sarawak. Please come over. Is he there?
This is
okay.
This is the award.
Yes.
He's not here. She's not here.
Okay.
She's not here.
So where is announcement? Okay. Okay, then the next is Amir Reza Emadifor from University de Sao Paulo. Be there or have just announced? Okay. Danting Wong, University of British Columbia. Yeah, come. She'll be here. You just.
They said we have to use this. The cash price is.
Next, the Best Presenter Awards, 3rd place. Junting Lee, a PhD student from Sisang ANF University. Professor? Jumping. Okay.
To give the award.
Okay. What's the name? Jumping. Jumping Tai. For this, I would like to invite Professor Chunting. Can just, use just stand here. Okay, Chunting P for he's Junting Lee. Okay. Yeah. So he'll present. Yeah.
I'll present this to you.
Okay, Jumping Tai. Okay, fine. Okay.
Yes. You stand in the middle. Yeah.
Then we have the 3rd place, Xiaowei Yi, master student. Please come. You have to be here. Okay, this is okay. 2nd place, invite Professor Yanjun Lee. Okay, this is the 3rd place.
I'll be here for a while because there's 8 of them.
So I will invite Professor Yanjun Lee.
Yeah.
Yeah, please.
Already?
Yeah.
Oh, okay. 2nd place.
Yeah. 2nd place is Hugh Lee. Okay, still 3rd place. Okay. 3rd place. Okay.
Okay.
Please 3rd place.
Okay, who's
Xiao Li?
Xiao Li.
Next
Okay,
one.
okay. This is online.
Hui Li. You can stand in the middle, maybe. Yeah, in the middle. Congratulations. And Yifan Xia.
3rd place. Yeah, okay. Do we have a 3rd place?
Master student. Yeah.
Is that you? Yeah.
Okay, let's go to the next. Muthusamy Ramakrishnan, Nanjing University. can stand in the middle. We are still Jinyu Liu. Zhen Yu from master students. Masjid Shirazi. Is he here? Masjid Siraji.
Yes, yeah, he's not. He's in Vancouver.
Okay, okay,
Yes,
okay.
yes,
So
yes.
we can maybe.
So you should go. That's right.
Okay, let's see. Okay, now. Thank you, Professor.
Okay, thank you.
So next is the Best Presentation Awards, second place. Wait. Yeah, I invite Professor Yanjin Lee now. Yes, please. Please do the honors for giving the award. Second place. Sing Swan Su. Okay, thank you. You'll be still here. Shanyu Pan, you are here now. You are going to receive it. Thank you. Answer, please. Here. Ming Peng Lee. Congratulations. Melissa Suganov. Melissa. From the University of Optics from Philippines. Second place. Yeah, another. Ayushman Malakar from Ranchi. Is he here? Is he here? From Ranchi, Malashkar. Ayushman Malakar. Okay. Okay, another second. Lucy Winfield from University of British Columbia. Online. Okay. Okay, then Menghong Xiang, ICBR. Menghong Jiang. She's not here? Okay. but not, I think she's not sitting in the hall. We go further ahead. Yanglin Xia. Yeling Xia. Not here? Okay. Presenter awards. Okay. I invite Professor Inns, please, to do the honors. Okay. First place, Pan Yang. Pan Yang. Let's see again, sir. There is another. Amsalu Alameriu from British Columbia. Okay. Do you have another? I'll just check. Yeah, there is. Tianhi Yu. Tianhi Hu. One more. Okay. One more. Yes. Xiangqi Wang from ICBR. Xiangqi Wang. Okay, she's not there. Okay. Oh, thank you all.
Okay, thank you all for attending the closing ceremony of our forum. We'd like to express our heartfelt gratitude to all of you who present, who attend, who gave discussions to all the forum. And yeah, thank you so much. And we will see you next year.